We're starting. To our speakers, remember to glance over at Federico and me, and we'll show you how much time you have left. So it's 5, 2, or 0 minutes. Our next speakers are going to be talking about small things related to saltwater and freshwater. Just as a little reminder, I'm sure you all know this, that Costa Rica's land mass is about 50,000 square kilometers. Costa Rica has domain over about 10 times that, so 500,000 square kilometers in the ocean. And these folks were brave enough to jump in and study things related to water. Our first speaker, Josie Taylor, will be talking about microbiomes of coral.
Thank you. Hi everyone, so my name is Josie, and I'll be talking about the microbiomes of Pastelpora coral in Costa Rica. So first up, I'll just talk about the importance of coral in our communities and the habitats of organisms. So for starters, they provide safety for coastline communities against storms. They provide habitats for many marine organisms. And because of that second point, they're a huge economic significance because they provide commercial and subsistence fisheries with jobs and also tourism. And so as many of you probably know, the coral life trajectory is not looking great for us. By 2050, it's estimated that 90% of all coral will be dead. But with recent research, it has been proven that there's huge importance with coral microbiomes in the fight against climate change, bleaching, and coral death. And so that leads me to my central question of how do Pastelpora microbiomes compare at varying health statuses? So my hypothesis was that as health declines in the coral, the microbiome diversity and richness will also decline. So I categorized the health by 3 categories: live, bleached, and dead. And my methods, I went to 4 different sites along the northwestern coast of Costa Rica, and I collected 3 samples from each site. 1 live, 1 bleached, 1 dead. I took them back to the lab, used a centrifuge to remove the tissue from the polyps. I then stained it with methylene blue dye to help differentiate between bacteria and different microbes. Then I looked at them at 1,000 magnification, and then I morphotyped the microbes. So here is a little example of how I morphotyped the microbes. So on the right side, you can see a key that I made, which differentiates the morphotypes by size, color, shape, grouping, and their extremities. And then I overlaid a graph here on all of my images to help determine size. So here we're looking at an image with a diameter of 178 microns and 10 quadrats across, so I can tell how big each of the bacteria are. So now for some results. So on this graph on the y-axis, we have species richness per micron squared. So I did it by area because I had varying amounts of data for different samples, so this way it accounts for that. So the blue is live, the orange represents bleached, and the gray is dead coral. There was a significant difference between live and bleached, and as well as live and dead species richness. Here's species abundance. Although it might look like there's a great difference between live and bleached, as I mentioned earlier, the y-axis is by pretty minuscule increments, so there was no statistical significance. And then I also measured microbial species evenness. And so what this means is on a scale from 0 to 1, how even the species are distributed within a sample. So considering they're all around 0.25, it tells us that these samples were dominated by just a few species rather than fully even. And then also to double check my methods and make sure that there was no differences in site collection that had an effect on my abundance or richness calculations, I ran an ANOVA test between all the sites regarding abundance and richness, and there was no significant difference found, which was great. The next thing I did was calculate alpha diversity index. So here I calculated two different sets of alpha diversity numbers. The first one was for live, bleach, and dead individually as groups. And then the second one, you can see where they overlap here, was a comparison of the two and their alpha diversity index of the two combined. The number in the center is the gamma number, so that's 113 species across my whole study. The next thing was beta diversity. So here the same thing, where it overlaps was comparing the two. So you can see dead has the highest beta diversity index, which means that it has the least similarity in species with the rest. So in conclusion, I found that live coral had the highest species richness, which supported my hypothesis, and the site locations had no significant effect on the data, which supports the reliable data that I provided. And then the third thing was a steady decline in species richness with respect to coral health. So as shown in the alpha diversity index, you could see as it went from live to bleach to dead, the species richness also declined. And then also there was a proportional decline in species similarities with respect to coral health, and that was shown by the beta diversity index. So this data shows that there's a slow loss of symbiont microbes after coral has expelled Symbiodinium, which is the symbiont algae that lives in the coral polyps. And if I were to expand on this study, I would track and ID the most abundant morphospecies throughout all the samples. I would compare to other coral species in the area of northwestern Costa Rica coast. And then also I would compare to other Postilopora samples from other sites outside of the sites that I collected from. And these results, the possibility of expanding this study can help in targeted restoration efforts to coral in Costa Rica. And lastly, I'd like to thank the Laras family for taking us out on the boat every day and night to collect all of our samples, and Doña Claudia for accommodating us with our crazy work schedules and staying up late and all of that. So, thank you.
Time for questions. Ready to go?
It's a very interesting way for categorize the bacteria. In the morphotypes that you found in the live corals, do you have a sense, or do you know, are these bacteria kind of detrimental, or are they positive in terms of the presence in this coral?
Have I identified the bacteria? Is that your question?
I'm asking, called my attention that you found in live coral. I am asking if you know about how positive or negative could be the presence of bacteria in live coral.
There has been studies that show that bacteria help significantly in their fight against harmful pathogens. But the bacteria I observed specifically, I'm not sure, I haven't identified it, but there has been research done that bacteria is very helpful.
Thank you.
Were you able to identify any morphotypes besides Symbiodinium in your coral samples?
No, not yet.
Do you think that they were there? You saw them, just not
The
able to?
Symbiodinium?
No, any additional bacteria or symbionts in the coral?
Yeah. There was a lot of bacteria.
Okay.
Yeah.
So you used a centrifuge to concentrate the bacteria, right?
Mhm.
How much of a difference do you think that made in being able to detect like the sample of bacteria from the coral sample?
I think it made a huge difference, especially for the bleached and the dead coral. The live coral, it was pretty easy to pick out the tissue from the polyps, but for the bleached and the dead, it's almost impossible to do that with tweezers. So, yeah.
So it definitely helped.
Yes.
Yeah. Is that considered standard procedure among people who look at microbiomes of coral? So
Yes, I read that in many papers. That's
Okay.
what they did.
Other questions? Muy bien. Thank you, Joseph.
Oh, thank you.
Our next speaker is Jessica Riker, who'll be talking about coral bleaching of Pocilopora.
Thank you. So I will be talking about Pocilopora, so let's get right into it. Pocilopora is a genus of coral and is sometimes referred to as cauliflower coral. It is found in the Indo-Pacific, Eastern Pacific, and sometimes the Indian Ocean. It is one of the first corals to bleach when temperatures change and is one of the worst at recovery after it bleaches. It has wart-like growths and is dome-shaped or branching. Coral is important because it provides a habitat to other animals like octopuses, oysters, sea stars, sea urchins, and many species of fish. If these animals decline in population along with the coral, food webs will alter, and top predators could decline in population as well. Coral bleaching occurs when water gets too warm or too cold, and the coral must let go of its symbiotic zooxanthellae living in the coral, and this causes the coral to turn worms. Coral can survive bleaching only for a few weeks before it either dies or recovers. Climate change has had a big impact on the coral. Climate change is the long-term changes in temperature and climate patterns due to human activities which release more carbon dioxide and methane into the atmosphere. One of the climate change patterns that has become more frequent and intense is El Nino, or El Nino Southern Oscillation. This event causes sea surface temperatures to increase in the Eastern Pacific, where a lot of the Pocilopora is found. This has led me to ask, what are the proportions of fully bleached white coral sections, partially bleached white and orange-brown coral sections, and total bleachness, fully bleached plus partially bleached coral sections of Passalacora in the Pacific coast of Costa Rica, and do size of coral, isolation, and water depth affect this? I would like to define partially bleached coral as the sections that are orange, brown, and white, shown in the photo. Fully bleached coral are the sections of coral that are completely white, and as you can see, a coral head, which is the entire coral, can have both partially bleached coral and fully bleached coral. And total bleaching is the combination of these two parts. And I would like to define isolation as the distance to the next closest coral. For my methods, I went to two sites, Baja Tomas and Baja Elachel. I used a transect tape to measure the depth of the coral and a seamstress tape to measure the size and isolation of the coral. I then used different size quadrats to measure to count the squares of partially bleached coral, the squares of fully bleached coral, and the total number of squares the quadrant filled up. After taking these measurements, at the end of each day, I took the temperature and pH as well. So this graph here shows the percentages on the bottom in different ranges and the number of coral heads found in each of these percentages for both sites. These graphs break down the last graph even more, so they show the partially bleached coral heads and the fully bleached coral, and they could be on the same coral head or on different coral heads. And based on these graphs, the data show that the total bleaching in the range 90 to 100% have 31 out of 50 corals, or 62% of the corals, which is a very significant amount. It also showed that in both bays, they have similar numbers of coral in the 0 to 15 and 90 to 100% range for the total bleaching pattern. Baja Tomas had more partially bleaching in the 0 to 60 range and more fully bleaching in the 70 to 100% range. And Elachel had more partial bleaching in the 70 to 90% range and more fully bleaching in the 0 to 15% range. This next graph shows the depth each coral head was found at against the percentage of total bleaching. And again, this is broken down, so it also shows the partially bleaching, full bleaching, and total bleaching in this graph. These graphs showed that Baja Tomas had depths between 220 to 340 centimeters, that many corals were 25 to 50% bleached, and this showed up for both partial bleaching and total bleaching. In the graphs, the tide was not taken into account, but if it was, Elachel would have been the deeper of the two sites. This next graph looks at the size of each coral head and the percentage of total bleachness. And once again, it is broken down so you can see the partial, fully, and total percentages as well. For Baja Tomas, it had many large-sized corals, and on Baja Elachel, it had mainly small-sized corals. This could be a factor as to why Baja Elachel had more total bleaching than Baja Tomas. Also worth noting is that Baja Tomas, its bleaching percentages between 20 and 60% were mainly medium size, and Elachel did not really have any within this range. This next graph looks at the isolation in response to the bleaching percentages, and this is the total percent bleaching across the two sites. And once more, this graph breaks down partially, fully, and total percentages. When looking at this graph, it's a little bit scrambled, so there was no difference in any category of percentages in relation to isolation for either site. To recap the data, depth data showed that depth could be a factor of why some coral was bleached, was more or less partially bleached than others. Size showed that the site with smaller corals had more overall total bleaching, and the isolations showed no difference in bleaching percentages. These factors matter because identifying which factors are related to coral survival will help in rehabilitation processes, which will give our coral the best chance of survival, which we know from Josie's presentation is very much needed. Coral farms are just one example where this data can be used, since they are the ones who plant coral. Different study ideas include going to a less bleached area and also studying a year that's not an El Nino year. With coral less bleached, the factors I looked at, I hypothesized might show more of a difference. And lastly, I would like to thank all the people who helped me. Leah, Haley, Rachel, and Josie all helped me collect my data in the field, so a big thank you to them. And I would also like to thank my advisors, Frank and Audrey, for their project suggestions and corrections, and another shout out to Frank for his picture rental. So thank you so much.
We have time for questions.
With the data that you specifically collected in your study, if you were to design a coral farm, what conditions would best suit the growth of coral based on this data?
Based on this data, I would go for larger size corals in clumps rather than spaced out. And I would also try and put it at a medium depth, just so they would have the best of both sides.
Okay, great.
Jessica, maybe I will read this in your final test. In terms of endangered or vulnerable or this concern, do you know about corals, where they are categorized? And I am curious, how many species of Pocillopora are there in Coyhaique, or is only one?
I could not tell you how many species
Okay,
there are,
maybe it's one.
but I believe it is in the not the critically or not the endangered category, the one right below. Although I would probably move it up one category or two, because they are like facing high rates due to global warming and temperature change.
So do you know if we can find corals in the red list of species?
You can
Maybe.
find certain, if I was looking just at genus, but if you go deep
Okay.
into species, you will find those in the.
Thank you.
Questions? Thank you, Jessica. What's up? Our next speaker is Lindsay Nielsen, and she's going to be telling us about acoustic landscapes or seascapes in coral reefs.
Okay, hi. I'm talking about soundscapes of coral reefs, and three different coral reefs, including keel. So coral reefs are very highly biodiverse, but very vulnerable habitats. They're very threatened by factors such as overfishing, tourism, pollution, and most notably climate change. Rising temperatures cause coral bleaching, meaning that the coral structures expel the zooxanthellae
algae living within the structures because the heat induces stress on the algae. The coral then turns white and dies. And they need the zooxanthellae to support the ecosystem, so when the algae is expelled, it leaves the reef very vulnerable and unhealthy, and it likely will die. Many organisms live in these habitats, so the coral reef is a very significant biologically diverse ecosystem. And these are some pictures of Poeciliflora genus coral. So soundscapes are a way of studying coral reefs. A soundscape is the ambient sound produced by the biotic factors in an ecosystem. In coral reefs, some of the sounds that we hear or that I picked up in the recordings are grazing organisms making scraping noises, or shrimps that make popping noises, which I feel like a lot of us hear when we're like snorkeling or swimming. You hear that like crackling noise. And the sounds that are produced by biotic organisms are called biophony. Studying soundscapes creates a more detailed, complete depiction of an ecosystem rather than just using visual observations. And it is a very non-invasive way to study organisms' behavior, as well as monitor the biodiversity in an ecosystem and the general health. To assess soundscapes, I used the Acoustic Complexity Index, or ACI, which is a metric that reports the biotic factors contributing to the soundscape. So when we were swimming and snorkeling around, as I said, we hear the like popping and crackling noise, which led me to the question of how different soundscapes and different reefs, or how do soundscapes differ between 3 different coral reefs of varying health levels. And I predicted that coral reefs with less bleaching would have higher acoustic complexity indexes, meaning that they would be more biodiverse and have more organisms and more sound. To do this study, I chose 3 reefs, which were Eucote, Elachal, and Octopus Garden. And then to quantify the reef health, I surveyed the reefs by swimming over the entirety of the reef, counting the amount of bleached coral colonies versus the amount of healthy coral colonies, and I was able to calculate a percentage, and then ranked these 3 reefs from least bleached to most bleached, which was Eucote at approximately 45% bleached, Elachal at 60% bleached, and Octopus Garden at 85% bleached. And then I placed a AudioMoth acoustic recording device at the center of each reef by attaching it to a weight and placing it on the ocean floor. I recorded 60-second intervals with 3 minutes in between, and I recorded for an hour at each site. Then with my recordings, I used an acoustic analysis software called Kaleidoscope to obtain the acoustic complexity index. And here I have my raw data. Each point is 1 recording at each site with the ACI that was recorded. And here we can see the trends in the ACI at each site. So I ran an ANOVA test between a one-way test, and I got a p-value of less than 0.0001, which my statistics are significantly different. There's differences between my 3 sites, but as we can see, Eucote had the lowest, and there's differences between all 3. Eucote had, which was the healthy reef with the least amount of bleaching, had the lowest ACI, which was surprising. I expected it to have the highest ACI, but this was likely due to the tides changing rather than the amount of bleached coral. I recorded at each site during different tides. I recorded at Eucote during high tide on November 26th, El Litoral during mid-tide on November 30th, and Octopus Garden during low tide on November 30th. And there is, based on my data, there's a very weak correlation between the amount of bleached coral and the ACI, and more of a connection between the varying tide levels. So to study this further in the future, I would use more coral reefs with vast health levels, and I would record over a more prolonged period of time to study the different sites at different tides. And yeah, thank you to my advisors and my peers for guidance and support.
Time for questions. Jake?
I'm just curious, did you happen to hear any other sounds from organisms other than herbivores or shrimp? Like, did you hear any fish like croaker or drums?
I don't think, no, not other than like grazing fish. Yeah, like gobies and stuff.
Do you have any ideas as to why changing tide level, specifically low tide, would yield higher acoustic complexity?
Because at different tide levels, there's just different organisms. Like the higher tides, there's going to be more fish, whereas like low tide, it's going to be more of the grazers and stuff. Yeah. Yes.
Was that the majority of the sounds you were hearing? Was like the fish that were using, or what was
the clicking, popping noise that you hear at like coral reefs is shrimps that make a snapping sound to stun their prey, and it's pretty frequent. And that's a general baseline for reef health in like soundscapes. That's a general baseline. Yeah, the shrimps. Yes.
So is that the crackling that you hear?
Yes.
What do urchins grazing? Do they make a sound?
It's more of like a scraping sound.
Okay. So the big sounds, for example, like an octopus garden or the shrimp.
Yes. Yeah, the crackling. Yeah.
Cool.
Yes.
What would you expect if you sample like let's say the 3 different coral reefs at the same time?
I don't know. I would be interested to see. I would think that my prediction would be closer to true if I were to do like all different tides at the different sites. I would think that Hikote would have the highest ACI if I recorded them all at high, middle, and low tides. I would think that it would have to do with bleaching, that a healthier reef would have a higher ACI. So Hikote would have a higher ACI.
Thank you, Lindsay. Our next speaker is Haley Bendorf, and she's going to be talking about water quality from different sources in Guatemala.
Okay, so as Frank just said, I'm Haley, and I looked into the difference in levels of water contaminants and minerals between various sources in Coquimiccio. So what inspired me to do this was, I don't know if you guys remember, but in the very beginning in Hotel Cax, Frank mentioned when he was talking about Coquimiccio that there was a problem with chronic kidney disease cases. So when I heard about this, I was really interested in it, and I wanted to know the cause, and I looked into it and saw that there is a significant increase in these cases in Guanacaste province. And when I was looking into what the cause was, I found it to be speculated that it was arsenic contamination exceeding 10 micrograms per liter of drinking water. And that paper that said that actually looked into all of the districts of Guanacaste, and they looked at the 421 aqueducts, and they examined them to see what the level of arsenic contamination was. And of that, they found that only 4% exhibited high exposure to arsenic, meaning that the districts that were still having an increase in kidney disease cases, they didn't have high arsenic contamination, suggesting that thought that arsenic contamination was the main cause wasn't the main cause. So this inspired me to really look into water quality and what makes up drinking water, the health effects of that, and I found the importance of regular testing, which ultimately led me to my central question, which is, do any minerals or water contaminants exceed the recommended exposure levels for drinking water in Coahuila? So, to do this, I took 5 samples in 1 liter plastic bottles, as pictured here, and then I then sent these samples into a lab in San Jose called Bioanalytica, and they underwent testing for level 2 and level 3. So level 2 was the minerals, and as you can see here, it's in Spanish, but that tested for aluminum, calcium, magnesium, iron, manganese, zinc, nitrates, chloride, fluoride, the strength. There's a lot, so I can continue reading them, but I won't. But as I go into depth later in the presentation, I'll speak with them more. So there's a picture again. How I collected these samples specifically was recommended by Bioanalytica, and what they recommended was to get 5 1-liter plastic bottles and make sure that it's the same plastic bottle throughout. Dump the water out. We didn't waste this water. It was drinking, just want to make that clear. And then after that, I took the bottle when I got to the source, rinsed it out with the source water, so there was no interference there. Filled it until it overflowed. Oh, I also rinsed out the cap. And then I capped it and labeled it with masking tape and Sharpie and a code, which you can see here. And then the samples were refrigerated before being sent to San Jose. Okay, so here is a map of Coahuila and the places where I collected the samples. So UCEAP is the organization that regulates and distributes the water in Coahuila. So basically how the water situation works there is they have a main tank where they mix various sources of water, including well water and water from the protected areas of Santa Rosa, which is right there on the map. And once it's inside this tank, they treat it with chlorine, and then they distribute the water to the various neighborhoods of Coahuila, which are pictured here. So this is, like I said, a map of where I took my samples. Let me try to use this thing. Okay. Here is the Marcielago neighborhood, and I took my samples specifically from the Marcielago sector of the ACG, and that represents the samples of water and the water that the neighborhood of Marcielago drinks. And then up here is the Muelle. So the Muelle was the only sample that wasn't used for drinking, and I used this as an extra level of comparison because the fishermen specifically say, hey, don't drink this water. The fishermen specifically say, hey, don't drink this water, so I wanted to see what was in it. And then right here is Casa Verde, where I'm sure you know where that is by now. It's Frank's house in Coahuila that we all worked at. And then here was Aguas Calientes. That's another neighborhood in Coahuila. And then up here is a sample I took to represent the source of the water before it went through. So this is the well source that goes into the main tank. And this source actually wasn't the UCEAP source because we weren't able to have access to that. So we just took a sample from a well that's about the same depth and right next to that well. And right like there, I'm shaking. Right there is where the well is that the UCEAP uses. So I did just explain these, but just to give more depth. So Casa Verde and Aguas Calientes is specifically water that is distributed by the UCEAP, the organization that distributes water. And both of these waters are treated with chlorine. Then this, like I said, is the well that represents the source. So this is kind of to represent the well that distributes the water, the well water that's distributed to these main neighborhoods. But I will say the family of Pablo Narvaez Hernandez, they do drink this water. It is untreated, as well as the Rio Marciélago water. It's also not treated. And then the way, like I said, was not used for drinking. Okay, so there were a lot of elements tested, like over 24. So I'm not gonna read and explain to you every single one. So I'm gonna explain the ones that were found. So calcium, like everything, all of these minerals, they're good for you in some moderation, but too much is bad for you. So when calcium is within the recommended exposure level, it's good for your bones and your teeth. But if there's too much, it's really bad for your digestive health. With magnesium is really helpful with muscle and nerve function. But if there's too much magnesium, a symptom is diarrhea, which I'm sure we've all experienced this trip. Next is hardness. And hardness is specifically calcium and magnesium ions. And what I found really interesting about this when I was looking into it is that if water is too hard, it is a health possibility. It's bad to get kidney stones. So that can be a health indication that the water is a little too hard. Fluoride, I'm sure you've heard of this. It prevents tooth decay, but also too much leads to dental and skeletal problems. Chloride is good for fluid balance of your body. Too much can lead to hyperchloremia. That's when there's just too much chloride concentration in your blood. And it also can lead to fluid retention. Manganese is good for overall for your health. There's lots of things it's good for. But if there's too much, it can lead to neurological problems. Potassium is good for like fluid regulation in your body and your blood pressure. But if there's too much, it does affect your heart muscles. That being said, if you don't already have kidney problems, potassium is really rapidly excreted throughout your body, so it's not likely that if there's too much potassium, it'll affect you a lot. Sodium is bolded. Do you guys see that? It's bolded. There's the reason. So that sodium also helps with fluid balance throughout your body. Too much of it, fluid retention, because your body's trying to put more water to compensate for the extra sodium. And if there's too much sodium, it's bad for your heart, your blood vessels, and your kidneys. This is a lot of stuff, so bear with me. Sulfate is good for your biological functions, but if there's too much, and also this is common if you drink water and you're not usually drinking water that's high in sulfate, you will also get diarrhea and dehydration. Zinc, good for your metabolism and health. If there's too much intestinal irritation, nitrates are good for physiological functions, specifically for your blood. But if there's too many nitrates, it can be converted to nitrites, which are really bad for you. And it can disrupt the way that your blood carries oxygen. And because of that, there's this disease called, I don't know if it's a disease. There's this problem that is called blue baby syndrome, and that's where the babies are born with not enough oxygen. So that's a sign that there's nitrates, and a lot of nitrites. And nitrates come from agricultural runoff. Okay, so wait, no. As you can see, there's a lot of things that are in water and drinking water that can go wrong. So I wonder what happened. Okay, so nothing really happened, which is good because that means that the water is safe and healthy. Here is a table of the results from level 2. Now, there's a lot here, so you're probably already not paying attention. So here we have the level 2 chemicals, and these are the minerals that can be good for you. And no aluminum was detected. I mean, you can go through these values, but the Decreto 38924, that's the recommended exposure regulations by the Costa Rican government. which is just making sure it's like, this is the healthy range. So nothing went out of the healthy range except, which one do you think it is? The red one, sodium, which was bolded. So that is kind of like that. If you're looking at that, that's kind of concerning because the recommended exposure is up to 200 milligrams per liter, but that's 349.41, which is a lot. So yeah, that's concerning. But that being said, mostly everything is within the range, and I just bolded the highest values here. Moving on to the scary one, which is heavy metals. What are you guys thinking? So nothing bad there, which is good. So yeah, the only one that was detected was the nitrates, which is from agricultural runoff, and I mean like, these are such low values compared to the exposure limit of 50. This is like all in the 0 point blah blah blahs, so like we're good. So yeah, okay. So to conclude and to discuss, okay. So as of November 22nd, 2023, which is when I took my sample, the only mineral or water contaminants present that was above the recommended exposure threshold by the Costa Rican regulations was sodium. And this was specifically in the untreated well source that isn't distributed. Oh, that is 0. So I should stop. But yeah, so just to summarize, that's it. Water quality is super important, and it's important to do frequent tests. And thank you to all of these people that helped me.
Time for one question. Is lunch here? Okay, awesome. Well, 2 questions. Jim.
So if you didn't like see any arsenic in the water, what do you think? Do you have any idea if it's causing it?
So, yeah, the kidney problems can be caused by a lot of things. There's a lot of health factors that can go into that. I was talking with Jordan a long time ago when I was like doing this, and we were just talking about how it can be not drinking enough water, like the sun exposure, the diet. There's so many factors besides this, but it's not the suggested cause that everyone was saying, which was arsenic contamination, as in the samples that I tested. Not for sure. Yeah.
What was specifically the source of the water in the Muelle? Like, was it from the tap or?
So it came from one of the spigots at the Muelle, and it wasn't really clear where that came from. I mean, do you remember?
It comes from a well between the Colegio and the farm themselves. They also pay.
So it comes between the farm.
Okay. And they said don't drink.
Yeah, and they said don't drink that. Frank was the one that gave me that idea to include.
If there were arsenic in your results, what would the source be? Would it be like the pipes or anything else?
That's kind of unclear. Everything is so specific and unique to every situation. So if there was arsenic, I mean, we'd have to do a lot more tests and a lot more interpretation to find out what exactly is causing that. So, yeah.
So we're going to break for lunch and start again at 1 o'clock. And very quickly, you'll hear a little more about this. Thank you and congratulations to this morning's speakers. Fantastic talks across the board. Thank you. And we look forward to talks in the afternoon. The speakers will be in lunch.
medicinal properties. And we're going to start with Emma Torres-Levine. And she's going to talk about mycorrhizae in a species of the basic called Cohoa cuscoicensis.
All right, I'm really excited to share my research I've been doing for the past month with you guys. So I was investigating the abundance of mycorrhizae on the species Cohoa costarricensis across an altitudinal gradient. All right, so nutrient management occurs because of microorganisms. And beneficial microorganisms live in symbiosis with plants across the entire forest, most of the entire forest. Mycorrhizae sprout from fungi, and they facilitate nutrient exchange by hyphae, and these hyphae act as pipelines between plants and fungi. However, this service is not free, and plants provide carbon and sugar to the fungi, while phosphorus is provided to plants by fungi. And this symbiosis system occurs in 70 to 90% of terrestrial plants across the forest. And you can see in this photo, this guy on the left has no fungi, so he's kind of small, and then the guy on the right, he's got some mycorrhizae, so it's pretty big. Definitely facilitates with growth. So what really interested me about this was it's very poorly understood where the system is distributed. So I was wondering how it's affected by increasing altitude, because there's a lot of evidence of the decrease of above ground net primary produce with increasing altitude. We're not sure exactly why this occurs, but some explanations are low photosynthesis rates because of a lot of cloud coverage, so plants aren't doing photosynthesis and they can't contribute to the exchange between plants and fungi through the mycorrhizae. Another explanation is low temperatures at the higher elevations, and one other explanation is root congestion, because there's a lot of water at higher elevations because of the clouds. So the water congests the roots in the soil. So I predicted, and mycorrhizae, the lack of mycorrhizae is also another explanation of, lower above ground net primary produce. So I predicted that there will be a higher abundance of arbuscular mycorrhizal fungi at lower elevations and vice versa. So my central question is, does the amount of arbuscular mycorrhizal fungi on the Cohoa costarricensis species vary across an elevational gradient? So the plant that I studied is this plant because it has mycorrhizal connections, as told to me by my trusted advisor. And it is naturally distributed. It's naturally distributed between 600 and 2,100 meters. And it's present in cold, humid areas, and it can be up to 15 meters tall. It was pretty easy to identify because it is alternate bipinnate leaves, and as you can see, the inflorescence is pretty bright colored with stringy red inflorescences. It's called angel's hair. That's the common name. So for my methods of answering this question, I collected root samples. I collected the plant at these 3 altitudinal gradients, and I collected 3 trees from each elevation and 2 root samples from each of the 3 trees at each elevation. And then I finally prepared and analyzed the roots. First, I cleared them. Well, I rinsed them first, and then I cleared them with potassium hydroxide, 10%. And then I dyed them to be able to see the mycorrhizae with 5% blue ink vinegar solution. If I didn't immediately clear it and dye it, I would store it with 2%
potassium hydroxide solution and put it in the fridge. Oh yeah, when I cleared it, I boiled the KOH, and I boiled the fluid vinegar. Then I cut the root pieces from the 2 root samples of each individual into 3 5-millimeter pieces, and I analyzed it with 400 and 100 magnification, and then I analyzed the pieces using a modified scale by Cormandic. And yeah, so this is the modified scale, the root colonization category. 1 means 0 is no mycorrhizae, 1 is 0 to 5% mycorrhizae. 2 means 6 to 25%, 3 means 26 to 50%, 4 means 51 to 75%, and 5 means 76 to 100%. And as you can see, there is a slight trend with a little more mycorrhizae. Yeah, these are each of the 3 individuals at 1,450 meters, 1,600 meters, 1,750 meters. There's a slight trend of more mycorrhizae in lower elevations, less at higher. However, there was no significant difference of this. So therefore, the arbuscular mycorrhizal fungi does not vary across an elevational gradient according to this graph from this way of quantifying it. I also quantified it another way using the average vesicle count. You can see a trend of definitely more mycorrhizae in lower elevations and less in higher elevations, and I did find a significant difference using an ANOVA test for both of them. I got a p-value of 0.0406, and yeah, therefore, this shows that the arbuscular mycorrhizal fungi does vary across the altitudinal gradient. So I didn't find a difference between the root colonization category at the different altitudes. This could be explained as the soil could have been similar across the altitudinal gradient, or the gradient I studied just wasn't big enough, like the sample size wasn't big enough. Yeah, so the abundance of mycorrhizae is the same across the gradient. And I also found a difference between the vesicle count at different altitudes, and this shows that the abundance of mycorrhizae varies across the gradient. This could be explained because there could be less nutrients at higher altitudes, as studies have shown. More cloud coverage, as you saw in the previous slide, which causes less photosynthesis, so less plants are able to exchange nutrients and contribute to that system, and water availability. So in all, nutrient transfer is an extremely important aspect of the ecosystem at work. And understanding nutrient transfer can help us understand how mycorrhizae and plants have adapted and survived to elevational changes over time. My hypothesis, to recap, my hypothesis stated that arbuscular mycorrhizae path abundance varies across an altitudinal gradient. The vesicle count was statistically significant in proving that, and however, my root colonization data was not statistically significant. Therefore, there's no conclusive evidence that there is less arbuscular mycorrhizae fungi at higher elevations than lower elevations, according to my data. So, yeah, I want to thank Naomi, Eladio, and everybody that helped make sure I made it down the mountain safe when I was gone for hours by myself.
You said there's less nutrients in higher altitudes, like those are some past studies. Do you know why?
That's a really good question. I'm actually not sure why, but they just took samples at higher elevations and a lot of samples at lower elevations of phosphorus, potassium, carbon, and there was a trend of a lot of lower nutrients at higher elevations, which I think could be a reason why mycorrhizae hasn't colonized at those higher elevations. Yeah. Hi, what's up?
Did you notice a given particular thing in the soil where these plants were growing in the different elevations?
Like, what do you mean particular thing?
What I'm thinking is, are these plants growing almost always in a flat place or well drainage, or is
They
there a
were
given
mostly
particular-
growing on hills, what I noticed.
Uh-huh.
I'm not sure if that has anything to do with it, but every time I took a sample of the plants, they were always on a slope, and the root kind of went down into the side. I didn't find any epiphytes. They were all just in the roots, but there are some epiphytic trees like this. Yeah, from this species. Hi.
Were the roots that you found more like herbaceous and fine, or were they like woodier and like thick?
Definitely more herbaceous and fine. Yeah, I actually found one epiphytic species, and it was just too hard to find the roots from that. I was like, no. But yeah, definitely more herbaceous.
Maybe because that's, I don't know, but why did you sample one increase the elevation?
Because it's a lot of like work and I didn't have enough time. I had tried to do another elevation, but it was too difficult to find it at the lower elevations. So I stuck with using 3 elevations, but I did want to use a bigger sample size.
But the trees were like difficult to find, or?
Yeah, it was pretty hard to find except at one elevation at 1600. But at 1750, yeah, it was harder to find it there. And it was also really scary because there was professional wasps at the higher elevation. So yeah. All right.
Our next speaker is Simposyum, and she's going to talk about mycorrhizae in fig trees.
Hi guys, I'm Lindsay, and I did my project on mycorrhizal fungi colonization at different stages of the ficus species life cycle. So to start off, we're talking a little bit about strangler figs, and strangler figs is a broad term for various hemiepiphytic species in the genus Ficus. They're distributed in the tropics and the subtropics, and the most common species of strangler fig in Costa Rica is Ficus aurea. They have 3 distinct developmental stages. They start off as epiphytic seedlings in the canopy, and then they grow their roots straight down to the forest floor, and that would be like the second stage. And then once their roots are in the forest floor, they wrap around their host tree, and it forms kind of like a trunk for the mature strangler fig. And they take over their host tree by physical constriction, blocking the host tree's access to light, and outcompeting for nutrients. And then they have, mycorrhizal fungi has a symbiotic association with plants by exchanging nutrients such as carbon, nitrogen, and phosphorus from the soil to their host tree's roots in exchange for photosynthetically fixed carbon. And past studies have found that ground-rooted ficus have more mycorrhizae in their roots than epiphytic ficuses. So knowing this and knowing what I know about the developmental stages of a strangler fig, my central question is, how does mycorrhizal fungi colonization vary during a strangler fig's life development? And so for my methods, I collected root samples from 6 epiphytic seedlings, 6 hanging hemiepiphytes, and 6 mature strangler figs. And for that, I had to climb some trees. So that was really fun. And then when I got all that, I went back to the lab at the Simposyum, and I stained them using, well, first I cleared them using 10% KOH, and then I stained them using 5% blue ink vinegar solution, and then I rinsed them with vinegar solution. And then I cut the roots into 3 5mm sections per individual. So in total there was 56 5mm pieces of root to analyze, and 18 were from each developmental stage. And so then I put them on some slides with water and cover slips. And then I used this scale to measure the percent colonization of mycorrhizae, and then pretty much how it would look. So like 1 is 0 to 5%, 2 is 6 to 25%, 3 is 26 to 50, 4 is 51 to 75, and 5 is 76 to 100. And then for my results, for the epiphytic seedlings, 2 were in the 0 to 5% category, 9 were in the 6 to 25% category, 4 were in the 6 to 50% category, 3 were in the 51 to 75% category, and the mean colonization was 2.44 with a standard deviation of 0.75. So that meant that most of the plants in this stage were in the 6 to 25% mycorrhizal colonization category. And that picture is from the assumed youngest plant that I collected from. It was like literally 2 leaves and some roots. It felt really bad ripping it out of its tree, but yeah, it had kind of a lot. It actually had the highest in the entire developmental stage with an average rating of 3.33. And then for the hanging hemiepiphytes, 3 were in the 6 to 25% category, 11 were in the 26 to 50% category, 4 were in the 51 to 75% category, and the mean colonization was 3.06 with a standard deviation of 0.39. So the average colonization was around 26 to 50% for mycorrhizae in this category. And that is a root sample from this developmental stage. And then for the mature strangler figs, 12 were in the 0 to 5% category, 6 were in the 6 to 25% category, and the mean colonization was 1.33 with a standard deviation of 0.37. And it's kind of hard to see in the slide, but there's not really much going on in that root. So. And then this is average mycorrhizal colonization by plant. And as you can see, the seedling stage has a lot more variation, while the other 2 stages are pretty even on what they are. And then this is the average mycorrhizal colonization by developmental stage. The mature trees have a noticeably lower average colonization level, and seedling and hanging roots are pretty similar, but hanging roots are slightly higher. And then I performed Wome ANOVA and found that there is a significant difference between these 2 stages with p-value of 0.0018, and then so I did 2 keys HSD and found that there is no significant difference between the seedling and hanging stage, but there was a significant difference between the hanging mature and the seedling immature stages. And then, so there was significantly less mycorrhizae in the mature strangler fig than in the other 2 stages, and I propose this could be, for the epiphytic seedlings, the most relevant abiotic constraint is water, so it would make sense that they'd have more mycorrhizae in order to enhance their ability to uptake water. And that also is supported by the fact that the youngest plant that I sampled from had some of the most mycorrhizae. And then for the hanging hemiepiphytes, they're also constrained by water, but when plants go from nutrient-rich epiphytic substrate to nutrient-poor ground soil, it would make sense that they would need more help from the mycorrhizae to uptake more phosphorus in the ground soil. So that's probably why that could be why there's more mycorrhizae in the hanging hemiepiphyte stage. And then for the mature strangler figs, once they've taken up their host tree, they don't really have much competition for nutrients, so they don't really need much mycorrhizae to help them. So that's probably why they have less. And then there was no significant difference between the epiphytic seedling and the hanging hemiepiphyte, but there was still a slightly higher average pollination in the hanging hemiepiphytes, which could be due to the fact that ground soil, like I said, has less phosphorus than the substrate that the epiphytes were growing from. And phosphorus is very important in strangler figs taking over the host tree from past studies. And yeah, in conclusion, mycorrhizal fungi is more prominent in earlier stages of strangler fig development, and it is suggested that lower phosphorus levels correlate with greater amounts of mycorrhizal fungi. And yeah, thank you to everyone.
Time for questions? Was there a wide range of distribution in which you sampled these figs?
So I sampled all the mature strangler fig roots and all of the hanging hemiepiphyte roots from the Institute, and then half of the seedlings were from the Gwyndon property and half were from the station, but there was no trend to show that the seedlings from the Gwyndon property and the seedlings from the station were different. They were all like pretty spread out in like both places.
So when the mycorrhizae colonizes the root, does it start in the substrate and then go to the root, or does it start on the root?
It can inoculate through spores or just like pre-existing in the roots or vegetative like hyphae. Just 3 different ways that it can inoculate into the root. Yeah.
Did you have to do different like clearing times and stuff for the different types of roots?
No, they were pretty similar. I stained the seedlings the most because they were the easiest for me to find, so I didn't really mess them up, then I could go get more. But they were all pretty similar in timing that it took.
If you have the chance to pursue basic studies, would you do something different?
I would definitely, like, while I do this part of the study, I'd also definitely take measurements of like phosphorus levels in the soil and in the substrate, as well as like pH, because past studies found that there's more mycorrhizae and like more acidic in lower phosphorus levels, because that's when they need more mycorrhizae. So I think if I were to include that into it, it would be more conclusive. Thank you.
Thank you.
Okay, our next speaker is Zoe Woodhouse, and she's going to talk about epiphytic burns on tree ferns.
Hi everybody, I'm Zoe, and my research project is on the elevational distribution of epiphytic ferns on tree ferns. So, tree ferns have existed for 300 million years, and they basically look the same since then. There's lots of like fossil evidence of that. And they were first present during the Carboniferous period. However, despite their persistence through time, knowledge of their general ecology is lacking relative to the knowledge of other like angiosperms and gymnosperms that we know a lot about. So, one thing that we do know though is of the 2 families of tree ferns that are in the tropics, one of them does have lots of like hair, but it's like it's roots that are both dead and alive, but they kind of have the appearance of hairs, and it creates this root mantle that has lots of spaces and traps a lot of nutrients, and makes a very good environment for epiphytes to populate. And so this picture is from a study, a study that was done. They took cross sections of a Permian-era petrified forest in Germany, and they found that the relationship between tree ferns and epiphytes existed since at least the Permian age, which is right after the Carboniferous period. And you can see these white arrows are all pointing to cross sections of epiphytic ferns on the fossilized slide of the tree fern. So I thought that was pretty interesting, and it just demonstrates how old this relationship is. And the forest was preserved by volcanic eruption. And it kind of led me to question, what is the relationship between tree ferns and their epiphytes? It's very clear that it's persisted throughout time. And more specifically, the relationship between epiphytic ferns and tree ferns. So in our study area, we're in a montane forest, and altitude is one of the important aspects of distribution since we have such a change in altitude. And in the forest behind the Simposyum, we have both primary and secondary forest. There's more disturbed areas, but there's also less disturbed areas. And as the path that I took, the principal path, disturbance kind of decreases with elevation. So I hypothesized that disturbance will impact the distribution of ferns along the trail, but I predicted that as elevation increases, so will the frequency and diversity of epiphytic ferns on the tree ferns, if we're going with the fact that it's less disturbed as you move upwards in elevation. And then this is just a picture to demonstrate like the hairs on the roots. This is not an epiphytic fern, but this is just the picture I had, so you can see. And then this actually is a picture of a tree fern with a lot of epiphytic ferns on it, and this is one of the tree ferns that inspired me to look into this relationship specifically. And it's just another picture of the rhizome. As for methods, I walked along the principal path behind the Simposyum, and then we start at an elevation of 1,540 meters, and then basically up to 1,800 meters. I marked tree ferns with flagging tape. Tree ferns in the tropics belong to either the family Dixonaceae or Cyatheaceae. And I would track all the morphotypes and take pictures of them 1 meter from the ground. And then I would count the amount of each morphotype that I found in each rhizome or trunk. And then I put that data into Excel. And then here's just some pictures of some of the morphotypes I found. I thought they were really pretty and fun, so I decided to include that. Okay, so in total, I looked at 28 tree ferns. I found 35 unique epiphytic morphotypes, and I had an elevational range of 243 meters. So my first graph was along an elevational gradient. I looked at the amount of morphotypes found on each tree just to see if there was any correlation on, maybe trees at lower elevation would have less morphotypes overall. There was no significant trend. And then I looked at the total number of epiphytes, since I had a head count of every single epiphyte morphotype, just to see if there's any trend with that. And once again, there was no significant correlation. And then, so some morphotypes did occur more often than others. This one I saw the most, and I actually saw this morphotype in San Gerardo as well. And most other morphotypes only occurred once. So I decided to look at the ones that occurred multiple times to see if there was any stronger correlation with that. So morphotype 5 was the picture on the leftmost part on the previous slide. There are some complete absences in some parts of the elevational range, but overall it tends to be present in most elevational ranges. And then This axis is just the proportion of presence, so out of all the trees that were in this elevational range, which proportion of them had morphotype 5 present on them. Same axes for morphotype 6, but this one actually shows a negative trend when it comes to elevation, which was different than all other ones that I found, but still not significant enough to prove direct correlation. Morphotype 15, once again, still not significant enough, but there was an interesting spike at lower elevation and then an absence until higher elevations. This morphotype, there was only, I think, 4 trees that had it, but it was the 4th most common morphotype, but it was only present in high elevation, so I thought that was very interesting and maybe could be something to potentially look into in the future. So, discussion. Trees were sampled along a trail, which trails tend to have kind of a higher rate of disturbance, more space in the canopy because there's a clear area where there are no plants growing. I also didn't have enough information to be able to confidently determine the families of each tree fern, but there could be differences in epiphyte distribution and occurrence based on the family, so that could definitely be something to look into. And since I found no correlation, I kind of think that maybe the diversity of epiphytes found might be a function of age more than elevation, because the older, or the longer that something has been present in the forest, the more time it has had to acquire epiphytes. So, but determining age was tricky, and with the scope and time frame I had, it was just not feasible. Yeah, elevations are the only indicator. The rest of the ecological factors are not thoroughly studied at all. I only found one other paper on epiphytes on tree ferns in the neotropic, or in Costa Rica, so that definitely should be something that we look into more. And just the relationship between tree ferns and their epiphytes in general. The rhizome trunks are a great place for epiphytes to amass because of the root mantles, as I described earlier. And in my personal experience, and the reason why I decided to go with epiphytic ferns instead of just like any epiphytes is they just seem to occur more often than all other types of epiphytes when, in my initial observations. So there could be something there. And because of the root mantle, there's actually been a couple studies done where they measured the circumference of a tree fern and then found a nearby angiosperm with a similar circumference and found that epiphytes did prefer tree ferns over angiosperms. Okay. In conclusion, there's a great diversity of 35 unique morphotypes, no correlation with elevation, and more research is needed to understand tree ferns and their ecologies in general. They are not the dominant vegetation, but they are still important to the forest understory. And it's always important to understand the biodiversity of our environments. Thank you to everyone here, my advisors, the staff at the Simposyum and the Monterey Institute, and the Delgado family for housing and feeding me during my research.
Questions for Zaddy?
Yes.
Was there one, I know you mentioned there was one more that you saw a lot more commonly, but was it mostly all completely different ones?
Yeah, besides the 4 that I showed, every other morphotype that I found was only present on one tree, which I thought was really interesting. Yeah, pretty diverse. Yes.
Have you, on your walks, did you notice, the birds growing on other, trees, or do they have, a preference for other trees?
The one that I kind of emphasized, I actually started looking at that specifically just because it interested me so much, and I think I only ever saw it growing on a non-tree fern. a couple times, but it was getting to the point where I would be walking and I'd see those epiphytes and I'd be like, I bet you that's a tree fern. I would look up and it was. So that's definitely something really interesting that I think someone else should explain.
Sorry, did you, I'm sorry you state this and I'm lost. Did you find tree ferns that are without other ferns growing on it?
I did. Yeah, I think there were one or two. I didn't mark those and use them just because I figured since I was looking for ones which epiphytic ferns, I would skip over those ones. But I think all of them had epiphytes of some
Most of them.
capacity, yeah.
So no any guess about why this didn't have ferns growing?
I think the one that I'm thinking of that I remember, it was in a very exposed part of the trail. So maybe it was a younger tree fern or there was more disturbance in that particular part of the trail. But, yeah.
Thank you, Zoe.
Our next speaker is Jenna Elman. She's going to talk about bryophyte diversity on big trees.
Okay. Hi everyone. So for my project, I proposed to look into bryophyte diversity and the increasing stratification of ficus species in Monteverde. Okay, so to start with, bryophytes are mosses, liverworts, and hornworts, and they tend to be epiphytes that flourish in lower temperatures with lots of water and light, which is why they're so prevalent in the cloud forests here in Monteverde. And they're super interesting because they vary by species based on elevations within the forest. But what's interesting about that is that there's a lot of research done on bryophytes and places we can reach, but once you go higher in elevation, it's not as feasible. So there's not as much research about what species exist at those elevations. So yeah, that's particularly where my interest was. Also studying bryophyte coverage moving up the Canby can serve as an environmental indicator of the impact the rise of the clouds in Monteverde is having on bryophyte coverage. And by understanding like the presence versus absence of bryophyte species, we can learn more about just the impact climate change is having. Okay, so this led me to my central question, which was how does bryophyte diversity within the increasing stratification of ficus species differ moving up the Canby? Okay, so for my methods, I chose 3 trees. One was at the Crandall and Dwight Memorial Reserve behind the Institute. The other one was at the Monteverde Friends School, and then the last one was at the Cleveland Family Farm, courtesy to Naomi. And yeah, so to do this, we had help from like professional tree climbers, and we would climb to the base of the crown. So for 2 out of the 3 trees, this was at around 12 meters, and then 1 tree was at 16 meters. And so we would get to the base of the crown, and then would hook a tape measure at like the highest point we climbed, and then rappel down, and then every 4 meters going down, I would stop, and then using a quadrant, gather the entire quadrant of bryophytes into a plastic bag, and then move down every 4 meters until I hit the ground. And then when I was done with that, I would do a densitometry recording and record the foliage coverage at that point in the tree. And then also for each tree, it was only on the northeast facing side of the tree in an attempt to standardize that. Yeah. And then once everything was collected, this was done at all 3 trees, went back to the lab, and then used my microscope to identify them by morpho species.
So yeah, for the 3 sites, they're all pretty similar elevations of about like 1450 meters, and the densitometer reporting was about was pretty much covered for all of them. And yeah, for the Monteverde Friends School, that one I was able to report up to 60 meters, and then the other 2 were both about. Okay, so for tree 1, this one had a total of 11 of the 18 morphospecies seen, and with only B1 at all sites. And this was actually the most diverse tree out of all 3 trees. And then tree 2 had 9 out of 18 morphospecies seen with B5 at all sites. And then tree 3 had 9 out of 18 again with B4, B5, and B12 at all sites. So this had the greatest overlap of species between all the heights reported. And then I compared the different morphospecies at each height as well. And so at 0 meters, all 3 shared B1 and B5 with 10 out of 18 seen. At 4 meters, B4 and B5, which is right here, were also shared. And then there were 11 out of 18 different morphospecies. And then at 8 meters, this was 8 out of 18 different species, morphospecies. And this one was the least diverse of all of the heights reported, and also had B5 as well, which B5 was seen across 0, 4, and 8 meters. And then finally for 12 meters, this was interesting. There was no overlap between all 3 trees, but there was the greatest diversity of morphospecies. So yeah, that was interesting. And then, yeah, so the overall results found 14 Bryophyta, which are mosses, 3 Mercantiophyta, which are hornworms, and then 1 Anthoceratophyta, which is a liverwort. Yeah, so basically, the goal was to measure the variability of morphospecies moving up in the canopy within the ficus species. But what was actually seen was that there was the greatest variability from tree to tree rather than by height, but this also could be due to the sample size, just because there were only 3 trees, and I think having more trees, the outcome might not be the same. Also, previous research has found a large proportion of bryophyte species within the inner crown that may be inner canopy specialists. And this coincided with my data because I found the greatest diversity of species at 12 meters, which was the highest point reported. And so this might indicate that some bryophytes only exist at these higher elevations in the canopy. But again, that's something that could use more research. In addition, something I wasn't able to focus on throughout the study was the weather patterns, but as you increase in height in the canopy, weather patterns change, specifically humidity, light, like water content, temperature, and bryophytes have very narrow moisture and temperature ranges, which means that different species tend to live in different types of other environments. So that'd be super interesting to see if you could standardize like the weather conditions while also looking at the different species. But overall, the study provided additional research about canopy bryophytes and the environmental indicators they can serve as. And with their relatively narrow moisture and temperature ranges, we can have a better idea of what conditions that they thrive in. And then using this, and also the knowledge of the rising cloud bank in Monteverde, we can record absence versus presence over time and get a better understanding of what exactly the impact of climate change and the increasing dryness of the Monteverde cloud forest is having on bryophyte coverage in all the microorganisms that rely on them. Yeah. That's all.
Questions for Jenna?
I thought when bryophytes reproduce, it's in the presence of water which carry the gametes downward. So, do you know how they get dispersed out of the tree?
I do not know that. No. Interesting.
What was it like climbing the tree? Were you scared of it?
It was pretty bad. We went with this guy who's pretty experienced, so felt pretty safe. It was cool. Lots
Why do you want to sample the northeast?
Just because I wasn't able to go all the way around the tree, and I wanted to standardize it somehow, based on like what weather conditions, like sunlight, as best as I could. Just because I wasn't able to do it otherwise, so using one side of the tree. Yeah.
Thank you, Jen. Okay, we're now moving into two presentations about bacterial inhibition. So our next speaker is Jake Willison, and he's going to talk about bacterial inhibition of plant and animal products.
I'm Jacob Willison. I'm testing the bacterial inhibition of varying plant remedies in animal products against Escherichia coli and Staphylococcus aureus. So about 75% of the approved antibiotics that we use every day are derived from natural products. Natural products are defined as anything produced by life, such as wood, milk, plants, coal. For example, penicillin is derived from a fungus, the most popular antibiotic used currently in the world. I mean, also, just out of the range of antibiotics, 50% of the world's medicine derives from natural products as well. I chose plants and animals to study specifically because plants contain beta-lactams, which are a secondary defense found in plants that kill bacteria as a defense mechanism. And plants are also everywhere in Costa Rica, so it was really easy to find them everywhere. It was easy to get help finding them as well. And I also chose animal products because some studies have shown that but probiotics, which are capable of fighting against harmful bacteria, are found in lots of many animal products, specifically yogurt, which in my experiment I used goat's milk and cow's milk, which are both used very commonly for yogurts. My question specifically was, what is the ability of various plant remedies and animal products to inhibit the growth of staph and E. coli? So to start, I obtained plants and different animal products from all over the place. I got some of mine from the Simposyum, such as like the guava, the dormignona, and the lemon. I got some from the Institute garden here, which Karen helped me find, which was really helpful because I didn't know where else to get them. I also sourced them from local homes and farms. Fede helped me find one, and I only got some plants and of the animal products from her family farm as well. For my plant remedies, they were turned into a tea. I got boiling water and I poured them over the leaves of the plants. Some of these teas included dried leaves, more specifically the dormignona and the lemon. They were dried beforehand because this is how it's traditionally prepared. And then some of the remedies also included juice from the fruit, more specifically the guava and the lemon as well. This is also because this is how they're traditionally prepared. And then the animal products were ready at the time of use, such as the honey and the milks. So for my experiment, I used the Mueller-Hinton-Agar plate method, which is the most commonly used plate method for antibiotic studying. Basically, the Mueller-Hinton-Agar plate serves as a medium for the bacteria to grow on. It's really common so the bacteria doesn't spread, and also it's just a good way of keeping everything contained and safe. So in my experiment, as you can see on the circles on the left, each circle represents a filter paper. I would dip the filter paper in my various remedies. Each color represents a different remedy that I used. For example, the yellow on the left is for the lemon, and those were used for both bacterias. And then these plates were repeated 3 times to keep consistency. And then I ended up doing about 24 plates in total because I had 16 products that I was studying specifically. And then these plates were put into an incubator for 18 hours at 37 degrees Celsius. And then I came back after 18 hours, checked on the bacteria, and then if they showed inhibition, they were measured, subtracting the filter paper, which is about 1/2 centimeter, and then that showed how big the ring of inhibition was. So in this one, the minuses represent if there was, the minus represents there was no inhibition, while the pluses represent full inhibition. For example, my regular honey and unpasteurized goat's milk on E. coli showed inhibition, And then the antibiotic eye drop was used as a control as well as water, which is used as a negative control. And then this graph represents the averages of the products that showed inhibition. So the honey was very successful at inhibiting E. coli, while the goat's milk was a lot less inhibitory. It only showed, out of the 3 trials against E. coli, it only showed it once, which brought the average down. And then just in comparison, the antibiotic eye drop was a lot higher. And then in my results against the staph, it was a very similar pattern. None of my plant remedies again had any inhibition against the staph. And then the only difference in this one was that the cow milk instead of the honey showed inhibition. So the goat's milk was consistently showing inhibition against both bacteria. The only issue with this again was that the goat's milk showed inhibition only one time, and then the cow's milk showed inhibition all 3 times, and only against staph. And then the antibiotic eye drop was slightly more successful against staph. So these plant remedies not showing inhibition doesn't mean that they don't contain any antibiotic products. A study showed that 31% of gymnosperm families and 45% of early angiosperm and magnolid families have antibiotic properties. As I mentioned earlier, 75% of modern antibiotics use plants. So these plant remedies could have just been prepared a little differently than normal, or I could have used more potent tea. I used about 250 milliliters of boiling water, and I could have brought that down a lot to get more of the juice extracted. Also could have done more of the leaves dried, used the bark from the leaf or from the tree, or crushed up the leaves. These are other methods that are commonly used for plant remedies, which I could have used. I expected the lemon and garlic specifically to show more inhibition because studies have shown that a low pH is very successful in killing bacteria, which from the acidity of the lemon, I expected to have more of an effect. And garlic contains a product known as allicin, which inhibits the enzymes that allow bacteria to grow, and I was expecting that to work as well. For the other plants, I was expecting those to work as well because of many traditional remedies use the plants I chose, and many past indexes have shown these plants having inhibitory effects. The unpasteurized goat's milk showing inhibition of both bacteria was expected. There's studies show that goat's milk contains lactic acid and hydrogen peroxide, which both have a low pH. And as I just mentioned before, low pHs are very successful at killing bacteria. The pasteurized cow's milk was partially expected. Many store-bought cow's milks that we buy today that are inorganic contain antibiotics from the cows, and then they're passed through in the milk. So I expected that to kind of work as its own form of an antibiotic. But since the cow's milk only worked against one of the bacteria, it was slightly unexpected. And then both the honeybee honey and then the mariola honey, which stems from a stingless bee, they were a bit unexpected because honey is said to have a low water activity and high sugar content. Both of those kind of work in hand. The low water activity, bacteria need water to grow, so by the honeys having a low water activity, therefore bacteria can't grow very well. And then the high sugar content of both works similar to that, as I said, where the high sugar content pushes out water due in osmosis. And then, yeah, acknowledgements, thank you for my advisors, and then the Institute for letting me use the lab and for helping me find all of the plants, and then for my homestay family as well. Thank you.
Questions for Jake?
Yeah.
I have 2 questions. One, you said you used Dormalina. When it was dried, was it like, were the leaves closed or were they open? And then the other question, did you look into at all, like, how have these methods been used at all, like, on a person? Like, I'm just curious, like, would you just, like, if you had staff, like, put goat's milk, like, on the infection?
Okay, so I think I meant, the Dormalina wasn't dried, it was actually the manzanilla. So that was separate. But the Dormalina was closed once you pick it off the tree, it does close up. And then for the other methods, specifically the mariola honey came in like a little bottle as like an eye drop bottle. So I thought that was interesting because that one didn't show any inhibition. And yeah, goat's milk can be drank. It can, like, as I said, the yogurt, it can be turned into yogurt, which has a lot of probiotics, which is helpful as well. Yeah.
Why was the goat's milk unpasteurized but the cow milk was pasteurized?
I was just picking 2, one pasteurized, one unpasteurized. It was also what I had access to. Yeah, as I just mentioned, I think it would have been useful to do the opposite of the pasteurization, just to kind of see if it had to do with the cow or the goat, because the pasteurization of the cow's milk did kind of make it have antibiotics and other special abilities.
Do you think that if you use like a different type of preparation concerning the plants, you would get like different results? For example, some people, like in literature, we know that some plants have some properties against certain conditions of birth. But sometimes you just took the leaves and you boil it like for hours.
Yeah.
Do you think that that kind of things can have an effect in the amount of antibiotics?
Yeah, I think, as I mentioned before, I think I could have used less boiling water. I used about 250 milliliters. I could have reduced that down to maybe 50 or 100, which would have had a more potent tea. Or even like crushing the leaves could have gotten different extracts from the leaves as well. So there is definitely different methods that I could use in the future.
Thank you, Jake. Our next speaker is Isabel Alvarez, and she's going to talk about other plants in addition to bacteria. Hello, everybody. So today I will be talking about
Ese técnico no está funcionando.
Hello, everybody. So today I will be talking about the bacterial growth inhibition of 7 neotropical plant species. So to introduce my topic, I will first define what antibacterial resistance is, why I chose each plant species, and how this topic is important, as well as my research question. So antibiotic resistance is when a bacterial species no longer responds to an antibiotic treatment that once worked to inhibit the growth of that bacteria species. This is a growing public health concern, as untreated bacterial infections can lead to septic shock as well as death. So for this experiment, I focused on 2 bacterial species. First, Escherichia coli, which infects 2.8 million people per year. This bacteria species has documented resistance against penicillins, aminopenicillins, and cephalosporins. My second bacteria species is Staphylococcus aureus, which is present in 30% of the human population at a given time, and this has documented resistance against penicillin. To explore plants as antibacterial agents, it is important to note that for hundreds of years, people have been using plant remedies to solve bacterial infections. And it is important to also note that in leaves, there are secondary metabolites that can show antibacterial properties. So for my plant species, I decided to focus on 7 different species
that are native to the neotropical region of Costa Rica, and some specifically only found in the cloud forest. So first, I have Miralana lobata. This is also known as gavilana. Bixa oriana, which is also known as achiote, and this is used to dye cheese and rice. Radiscanthia zebrina, this is also called hoja de milagro because of its medicinal use all throughout Central America. Ocotea monteverdense, which is only found in the region of Monteverde. Hyper species, Hamelia patens, and capsaicin species. My research question is, how much growth inhibition will occur in Escherichia coli and Staphylococcus aureus with 18 different plant preparations? To introduce my methodology, I want to first talk about how I prepared each plant, how I prepared bacteria, what a Kirby-Bauer assay is, and how I collected data. So in total, I surveyed 7 species of plants through crushed leaves. This included 6 grams of crushed leaves, and then I surveyed 7 different species of plants through fresh leaf tea, which included 3 grams of fresh leaves into 250 milliliters of distilled water. And then I explored 3 species of plants in a 48-hour dried leaf tea, which was 3 grams of dried leaves in 250 milliliters of distilled water. And there was only 1 species which I explored through a root infusion, which was 3 grams of roots of the plant in 250 milliliters of distilled water. And the reason why I had different numbers for each preparation is because I looked through the literature of historical medicinal uses of each plant species, and that's what guided me to prepare the plants in such a way. However, if there was no documented medicinal preparations, I then explored crushed leaves, fresh leaves, and a dried leaf tea. And this allowed me to survey what would be the best remedy for the undescribed plants. So for my bacterial preparation, I first made bacterial agar plates using a Mueller-Hinton agar mix. And both of my bacterial cultures were inoculated on the 11th of November in San Jose. And I then diluted the bacterial cultures in distilled water before spreading them onto my plates. So in a Kirby-Bauer assay, there's an agar plate which is divided into 4 quadrants. Each white circle is representative of a filter paper that is dipped in each treatment. And in total, I made 36 agar plates. I had a total of 144 tests, and this included 72 tests for each bacteria species. So after 18 to 24 hours, after 18 to 24 hours of bacterial incubation, you can then see there will be bacterial growth, and if the treatment is successful, there will be a ring of bacterial growth inhibition next to the filter paper. In order to collect the data, I then measured the distance from the filter paper to the end of the ring of inhibition, and that is the data that I will be presenting today. So I will be discussing all of the bacterial growth inhibition that I found, as well as the differences between each bacterial species. So for E. coli, there were only 3 plant preparations that were at least minimally effective against its growth. First, T. zebrina dried leaf tea, capsaicin crushed fruit, and O. monteverdensis dried leaf tea. On the right, you can see my positive control, which is antibiotic eye drops. For S. aureus, there were 7 species that were at least minimally effective against the bacterial growth of S. aureus. This included T. zebrina dried leaf tea, Hyper species dried leaf tea, B. oriana fresh leaf tea, B. oriana root infusion, capsaicin fresh leaf tea. There was only 1 plant preparation that was moderately effective, which was H. patens fresh leaf tea, and there was only one plant preparation that was significantly effective against the growth of staph, which was T. zebrina crushed leaves. I want to conclude that although I did find 9 minimal plant preparations, this does not mean that they are only just minimally effective. These preparations were done very minimally invasive. I just either prepared teas or crushed leaves. So if I were to further concentrate or further look at the chemical composition of these plants, I could yield very different results with these plant species. Additionally, with the moderate bacterial inhibition, which was just Hamelia patens fresh leaf tea, this was only moderately effective against staph. So that was the only plant preparation that showed moderately effective against any type of bacteria. My last plant preparation was Travescantia zebrina crushed leaves. This was the only plant preparation in all of my study which showed significant inhibition with just minimal preparation, which was crushing the leaves. And this showed over 0.5 centimeters of bacterial growth inhibition by just crushing the leaves against staph. So what does this tell us about the use of plants as potential antibacterial agents? First, that plants have extreme potential to be used medicinally as well as further developed into pharmaceutical products, as well as the fact that many future pharmaceuticals can be derived from plants, specifically in ecosystems that are under researched. And I want to further emphasize the fact that this research correlates with the fact that the cloud forest ecosystem is extremely important. So much of it is under research, and so the fact that it is endangered each and every day from climate change is extremely saddening for the scientific community. I want to also state that for future applications, many different plant preparations can be explored for like future students and or future studies. So that can be something that will be further explored, as well as the fact that the plant T. zebrina is the only plant from my entire study that showed bacterial growth inhibition for both E. coli and staph. Although one was minimally effective and one was significantly effective, this was the only plant that showed that correlation, which means that it could be a very good potential general antibiotic agent. Additionally, if I were to have more time, I would concentrate the active molecules within these plants, but unfortunately I was not able to do that. And I just want to acknowledge my primary advisor, Naomi, as well as Federico, they both helped me obtain a lot of these plant samples, as well as Edmida Porras, who allowed me to visit her farm, and it's where I got most of my plant samples. So I'm really thankful for also the Monteverde Institute for letting me use their lab. Thank you. It was Travescantia zebrina against staph.
Okay. Thank you. Yes? Do you have an idea of, say you did this study and you were able to have more time, or do it against different infections other than staph or like E. coli? What are some other things that you might test the T. zebrina against?
I would say that because of the fact that it showed at least minimal bacterial inhibition on E. coli and staph, I would do a whole array of like common human pathogens that cause bacterial infections, because of the fact that E. coli and staph are very different, and that's why I chose them both, so that I can see like if a plant was specifically able to treat one or the other. So I think I would treat it against a multitude of human pathogens, specific bacterial pathogens. Yes?
Did you taste any of the T. zebrina
noxious. They're very strong in scent. So unless I've seen literature that suggests that it would be safe to drink, I would not.
Do you know which can be the substance present in Nicebrina?
I am not sure. Unfortunately, I wasn't able to look at further research. I know that different literature looks at specific compounds, but I wasn't able to find one that expressed that crushed leaves would specifically have a greater result, so I wasn't able to.
I know that almost all the plants you use, they have a given knowledge about people using it. But I would like to know, why did you select to use Ocotea monteverdensis, which is unknown?
So there were two species that were unknown that I did not find in any literature, which was Ocotea as well as Piper. And the reason why I chose both of those specific species was because there's literature that suggests that the genuses Ocotea and Piper have antibacterial properties amongst all of the species in those genuses. And since Ocotea monteverdensis is an endemic species of this region, I really wanted to see if it had any antibacterial properties. Thank you, Isabel.
We're gonna take a break and come back in, did you say 12 minutes? 12 minutes.
coastal habitats, understanding how anthropogenic factors influence wildlife, like bats, becomes imperative. So different land use changes influences bat presence and activity differently. There's differences of adaptability and vulnerability among bat species, and this shapes bat populations. And for clarification, insectivorous bats utilize echolocation calls to forage for insects. So these are just a couple of the human-altered landscapes. The first picture is of an urban landscape, the second picture of a forest landscape, and the third picture is a forest-town interface, which is an interesting landscape because it's where the forest and the town meet. This is an important transitional area for many species, including bats. So my question becomes, how do different human-altered landscapes affect insectivorous bat diversity and activity? So my research method, I decided to use an audio map or bioacoustic tools like Kaleidoscope. The first picture shows the echolocation call of a myotis, and I decided to monitor 96 hours of acoustic monitoring with a total of 576 minutes recorded, and this was in a span of 8 nights. I chose each site depending on where it was. So this was a site at an urban area, and there was 4 urban sites, so 4 nights that were monitored. And for the forest town interface, I decided to place the audio mounts on pastures because a lot of the within the forest town interfaces, there's a lot of pastures and they're important ecosystems in which a lot of plants may exist. And so what did I find in the pasture? So these are cute creatures, or the species of bats that were found in the pasture, so a total of 5 species. There's a total of 23 bat passes. And for the urban area, these are the species that were found in the urban area, and there was a total of 9 bat passes. So, now this graph here shows the mean of bat passes per site. For the pasture, it's significantly higher than the urban, with a mean of 5.75. And the urban had a mean of 2.25. And the standard deviation for the pasture was very high, meaning that there was high variability within each night. Despite there being more bat passes in pastures, the p-value was of 0.021, which suggests that this difference did not reach statistical significance. So why are there higher bat diversity and activity in pastures? There are many reasons for this. There's a study that was conducted in Tanzania by Thomas Kazunzi that showed that there was actually higher species richness and activity in agricultural areas than forest areas and urban areas, which is interesting, but this is most likely due because there's higher prey availability and open spaces, which make it easier for bats, some species of bats, to forage. And there's also different species present in pastures than in urban areas. They only share one bat species, which is the Myotis nigricans. And then the molossus species was only present in urban areas. And something interesting about this specific species is that they're an urban exploiter, meaning that they tend to prefer urban areas than other landscapes. And this is because they have low predator avoidance behavior, so they do not mind light. They actually like street lights because insects tend to aggregate within street lights, and they forage in those areas, and they use urban structures for their rooting sites. Also, something interesting about urban exploiters is that their morphology, they tend to be larger in size and tend to be able to travel
larger distances. Urban avoiders tend to be smaller and forage within the forest. And so some factors that may have influenced my results is that 4 out of the 8 nights there was rainfall, and light rainfall doesn't really affect like insect behavior, so it doesn't really affect, consequently it doesn't affect bat behavior. But heavy rainfall does affect their behavior, like it makes it more difficult to fly, insects are not out as much, so they tend to not come out during heavy rainfall. Also moonlight, there was a full moon and intense moonlight, I think 2 out of 4 nights, and for some species of bats they avoid intense moonlight, like the Myotis species. They tend to not like being in intense moonlight because of predators. And so in conclusion, it's important to create awareness of the impact that land development has on the environment, more specifically on bat diversity and activity. Bats that are urban avoiders are vulnerable to land use changes, so it's important to look into that and see how we can help protect them as well. So I want to say thank you to my physical advisor, Andy, and to all the instructors and everyone here.
Opening time for questions.
I have 2 questions. The one is, what kinds of predators are there for bats in which they might avoid moonlight? And then the second one was, what was your p-value again? Because I thought that it was significant.
It wasn't significant. It was 0.021. Maybe I put it wrong. I don't know. But I think it's not significant. The difference itself.
Gotcha.
Yeah.
Oh, and then the predator.
Oh, the predator. But I'm sure there's.
Yeah.
It could just be like a behavior that they have.
Yeah, Stephanie, I didn't hear the whole, Stephanie, I didn't hear the whole explanation, but I had the same reaction. That p-value at 0.02, unless you're being very stringent, most of us would consider that either significant or slightly significant.
So I used the website, but I will have to check. And like the website itself said like it's not significant, but maybe I should have looked further on that.
Okay.
I'll check my Excel sheet. I could have probably placed it wrong.
If you said the p-value of 0.01, then indeed you would perhaps say that that's not significant. But I would definitely take a closer look at that. Yeah.
Gracias. Okay. Okay, next speaker from UC Santa Barbara, Maya Romero. She will speak about compositional comparison of ectoparasites of frugivorous bats along human disturbance gradient.
As Fede said, the title of my project is the compositional comparison of ectoparasites on frugivorous Phalostomidae along a human disturbance gradient. Basically just I measured the number of ectoparasites and the population of bats in 4 locations. Okay, so to begin, frugivorous means that an organism will eat fruit, and Phalostomidae is a common family in Costa Rica and is also native to the New World. I became interested in frugivorous Phalostomidae because they act as seed dispersers in ecosystems.
really cute. So an important part of this project was pesticide impacts. So sustainable farming is becoming much more common across Costa Rica, but still farmers are using pesticides to create enough yield for their consumers as well as just to consume the food they grow. With that being said, bats do live in agricultural areas, and it has been researched previously that bats could be used as bioindicators for the health of the ecosystem, as well as ecosystem functionality. Finally, ectoparasites live on the fur of bats, or in the armpit, or under the wing, and I use ectoparasites as a proxy to determine bat health, and in turn, bat health might determine ecosystem. So, taking all of those factors previously mentioned together, I began thinking about how ectoparasites and their abundance could be indicative of bat health and in turn ecosystem health, which led me to my central question of what is the impact of human disturbance of frugivorous bat ectoparasites along a human disturbance gradient. I also hypothesized that due to higher stress, chemicals, livestock, and increased human disturbance, that in highly disturbed sites there would be more ectoparasites. Not necessarily indicative of bat population. So I focused on 3 genera: Sternera, Artibaeus, Corolla. All are distinct with their leaf nose shape, but they have different characteristics among the genera, but they all do consume fruit. So for my methods, we sampled for 9 different nights, roughly 5:30 to. It depended on the night because 2 of the 4 nights were rained out at one site, but we nest netted from 5:30 to 8:30, 9:30, and then I based my data off of 4 sites. So the first site was Life Monteverde. I deemed this the highly disturbed site because it is right next to a road. There's livestock, a lot of human presence throughout the day. There's a lot of lights to maintain the livestock, and as well as noise pollution from the livestock. My second location was here, so that was an intermediate to high disturbance area because there's a lot of people present throughout the week, and it is in a suburban area compared to the other 2 sites that I will mention. So it was high and intermediate. And then my third site was Simposyum, Monteverde. It's half a mile up from a main highway in Monteverde, as well as it borders an intact forest. And then our final site that I used was Simposyum San Gerardo because it's a really big hike-in and very few people cross throughout the day, and less disturbance is logical in that area. As I mentioned before, there were 3 studied genera: Sternera, Artibaeus, and Corolla. And then the ectoparasites families that I focused on was Nectariidae, Strepsilidae, which are both bat flies, and then there is also Macrocinidae and Spintricidae, which are mites. I also removed the ectoparasites with tweezers, but a lot of times Bede and Stephanie had to use the fingers to grab the ectoparasites because they had wings. Okay, so these are some pictures of the ectoparasites I collected. The first, these guys, super long legs and very distinct bodies compared to the rest of the families. They were the most common ectoparasites to be found on frugivorous bats. The second family is Strepsilidae. They're diverse within the family, but we caught most commonly these 2 species. I'm unsure of the species. It's really hard to identify them down to the species, but they have wings, or modified wings, 6 legs, and distinct 3 segmented parts. Finally, we caught mites. So this first guy, right here, as well as the second guy, they have similar legs, but their bodies are different, and they actually do not have segmented parts. This one has like a weird, like translucent behind. They're pretty nice, but they're also really cool. So that was the focus. Originally, I wanted to see if it was a genera-specific relationships on
and 92 ectoparasites. I really want to discuss Sternera because we caught not only the most number of Sternera in this study, but we also caught the most number of ectoparasites on Sternera, which makes sense, which you will see. But we also caught 13 Ortobaius, 7 Corolla, so Corolla was the least number of genera caught. And then I also averaged ectoparasites per individual. So Sternera had roughly 2 ectoparasites per bat collected, and Corolla had the least amount, which was less than 1. Okay, so the following 3 graphs are very similar, but they are broken up by genus. So on the x-axis, I have the site. So there were 4 sites, like I said. And then on the y-axis, there was the number of samples. So it was both the number of bats and the ectoparasites. And I'm just gonna show you all 3 genuses first and then discuss. Because visually you can see that along the gradient, life being the highly disturbed and then Simposyum being the least disturbed, both ectoparasites and bats increased the farther away from human disturbance that we had. Based on the data, even when the farm was integrated like life with, or intercropped with trees and crops, this disturbed system was still not conducive for bat habitats. Only 4 fruit bats were caught at life and 2 at institute, which were both the least highly disturbed areas. So not only the bat population at these sites were low, but coinciding ectoparasite populations were also low. And this was true for all 3 genera. Interestingly enough, Corolla was rather elusive, but we did catch one at life, and it did have an ectoparasite. But there were quite a few more at Estacio and San Gerardo. It's unclear whether, because we were rained out 2 of the nights at life, even though the catching effort was really high, it's hard to tell what reason that is. We don't know. I wasn't sure if it's because there were bats present, but they weren't flying into the net. There were just no bats present, or if it was due to other factors, chemically, livestock, lights, rain. What I can discuss is the clear trend in bat population and ectoparasite population as we get farther away from humans. To explain, at least for Sternera, due to the high number, I read a paper discussing how bat flies don't transmit they transmit between bats, but they don't transmit while they're flying, they transmit within the roost. So an increase in bat population would likely mean an increase. Given a certain area, there's likely to be more roosts, therefore more Sternera, and therefore if the bat ectoparasite crawls up into the roost, they transmit way quicker. So if there's an increase in population, there's also an increase in ectoparasite population. Yes. And then, to conclude, Frugivorous Phyllostominae activity and ectoparasite load both increased were high in lowly disturbed areas. Again, I'm unsure if it's due to chemicals or other disturbance factors, but probably most importantly, I would like to mention that my study describes the importance of maintaining intact forests as well as doing our best to intercrop in farms even when chemicals aren't present. So I'd just like to thank Fede, Adri, and Emmy for helping me with the study, my fellow ectoparasite enthusiasts, and the back crew for helping me grab the ectoparasites. I literally would not have been able to do it without 4 or 5 people there. And my host family for always meeting our team and asking how my day went, and then all 4 locations that we samped at.
We have time for questions.
Are there any differences in roosting behavior difference in ectoparasite per bat from Sternia and the one that started with a C at Simposyum? Yeah, it seemed like there were a lot more per bat on Sternia. So I was wondering, or do they like have increased roosting behavior in comparison to others genera?
That I'm unsure about still. I know that from the research that I did, I know that females roost together and males are more solitary. I don't know if it's genera specific, although I do know for your respects do tend to roost similarly, but they do roost within the genera. I believe I'm correct on that. But yeah, I'm not really sure about like relationships amongst like the exact parasite and the exact genus.
Gotcha. Is there a way to easily tell the female and male? Did you know like when?
With bats?
Yeah.
Yeah, it's pretty obvious.
Okay.
You just look on their stomach and
well, yeah,
it's exposed. So
okay.
yeah.
Any other question? Gracias, Maya. Mari, could you come? So please remember that we are a friendly audiencia. Calm down, breathe. Sometimes look at me because I'm having some papers to tell you about the time. Bueno, thank you. Sorry for the technical mistake. Next speaker from UC Los Angeles, Mari Ogden. Is going to let us understand what factors influence photoparasite appearance in bats. Thank you.
On understanding what factors influence ectoparasite occurrence on chiropterans in the Monteverde region of Costa Rica. So the first thing that I wanted to go over are just some key terms. First being chiropteran. Chiropteran is the order of bats, and order is one way that taxonomists classify animals according to similarities that they have. The next is ectoparasites, which after Maya's presentation don't need much of an introduction, but they're just parasites that live on the outside of the animal's body. The next two terms that I wanted to talk about are inter and intraspecific. Inter means differences, or inter means between members of different species, whereas intra means between members of the same species. And lastly, I wanted to go over taxonomic classification. Again, this is a way that taxonomists classify organisms according to different similarities that they have. And the ones that I'm going to be focusing on from most broad to most specific are order, family, genus, and species. So there are a lot of misconceptions about bats. actually asked me if it was too late to change. So, the first thing is that bats are blind, and there's a common saying, "You're blind as a bat." And while bats can't see very well in color, they do have exceptional vision and can see in conditions that we would interpret as pitch black. The next thing is the relationship between bats and vampires, which is seen in anything from ancient folklore to modern film. Only 3 out of the 1,300 species of bats feed solely on blood, and all 3 of those species feed solely from the blood of animals, never humans. Next is rabies. Bats do have rabies, which is why you should never hold them with your bare hands, but only 0.5% of the world's wild bat population has rabies. The reality of the situation is that bats are amazing pollinators. They pollinate over 500 species of plants, including mangoes, bananas, and guavas. They're also the sole pollinators of agave, which means that we have bats to thank for tequila. A lot of bats also eat fruit, which means that they're integral seed dispersers. So when they excrete the seeds in their feces, they're able to disperse it over wide areas because they can fly. The other thing that they're really helpful in is insect population control. Research has shown that over 50% of insectivorous bats research has shown that insectivorous bats can eat up to 50% of their body weight in insects every night. The next group that I'll be talking about that's included in my study are ectoparasites. Bats can have any ectoparasites from ticks to mites to, in the middle you see, are bat flies. Why did I choose to do my study in Monteverde? It's because Monteverde has a very unique type of ecosystem, which is a cloud forest. Cloud forests account for less than 1% of global landmass, but they account for 15% of global biodiversity. And in particular, the Monteverde Cloud Forest Reserve has 2.5% of the world's global biodiversity, which is crazy when you think about how small it is. And this extends also to bats. Over half of the 100 species of mammal that are in Monteverde are bats. However, there's currently no data available on ectoparasite and bat relationships in this region. Which leads me into my central question. What factors play a significant role in ectoparasite occurrence on bats in the Monteverde region? To do this, I looked at interspecific variations, such as genus and diet, and also intraspecific variations, such as mass, which we got using this device right here, which is a Pasola scale, sex, which we got from looking at external genitalia, and also in the case of breeding females, mammary glands, which you can see under their wing, forearm length, which is this bone right here, and then also age. To get age, we would outstretch the wing of the bat like it's shown in this picture, and we would look at the joints, which you can see here and here, for example. And if the joint wasn't fused yet, you would be able to see the light through it, and we would classify it as a juvenile. And if it was fused, you wouldn't be able to see the light, and it would be an adult. And if it was kind of in between, then it was a subadult. So this study was done over 9 nights in 5 different locations around the Monteverde region of Costa Rica. The first thing that we did was misnet the bats and then put them in cloth bags as they waited to be processed. Processing an animal means to take all of the data that you wish to include in your study. And part of this, for Maya and I's study, was to look at ectoparasites, which we counted for each bat. In total, we collected 130 ectoparasites from 4 different families, including Nectaribidae and Strepilidae, which are 2 types of parasitic bat fly, and Spintornicidae and Macrocinidae, which are 2 types of parasitic mite. We also caught 76 bats from 2 different families, 8 genera, and 12 species. They're really cute, Caiguanis. The one on the left is Vespertilionidae, and the 2 on the right are part of the leaf nose family, also known as Phyllostomidae, and you can tell from this little flap of skin, which is like a leaf on their nose. In order to do statistical analyses, I did chi-square test using a 0.1 significance value. You can see on the left that this is the number of individuals relative with and without parasites relative to their forearm range, and the one on the right is just that data reflected in percentages. And you can see specifically the bats in the 40.1 to 44 millimeter range are had the highest amount of occurrence of ectoparasites. Forearm length was found to be the only interspecific variation that had any statistical significance in relation to ectoparasite occurrence. Genus was the only interspecific variation that had to do with ectoparasite occurrence. And although we found 8 genera, I only decided to include 5 in my data because the last 3 only had 1 individual each from each of the genera. And I'll talk about why this is a problem in my study limitations. The graph on the left, again, shows the amount of individuals with and without ectoparasites, this time in relation to genera, and the one on the right shows this information in percentage. You can see that the Sternera genus has the highest amount of ectoparasite occurrences. So, one thing that scientists think, and why forearm length might have been found to be significant, is that a larger forearm length means a larger wing surface area, which could mean that there are more room for ectoparasites to live in. This has been found to be true in a variety of other taxa, including seabirds and flying squirrels. But the thing is, that I mentioned earlier, is that forearm length, or maybe I didn't mention earlier, I'm kind of nervous. Forearm length is used to determine genera and species. If larger forearm length is indeed correlated with a larger surface area and more room for ectoparasites to live, my graph would have looked like that. But instead, my data looked like that. You can see that the forearm length with the largest amount of ectoparasites is 40.1 to 44 millimeters, which 30 out of 35 of the members in this size range were of the Sternera genus. So could genera-specific ectoparasite host interactions be the reason for the forearm length correlation? This leads me to discuss coevolution and evolution in general. Bats and ectoparasites have been around for up to 600 million years, and bats have been around for 50 million years, which means that they've had a lot of time to co-evolve with each other. It can also have something to do with roosting behavior. Some bats live in caves, which can support very large communities, whereas some only live in leaf tents, which support smaller communities, and some live alone altogether. Could coevolution between bats and ectoparasites be a factor in ectoparasite occurrence? One study limitation that I had is that the vast majority of the bats that we caught were adult frugivorous bats that were in the lower middle weight range of the sample population, with forearm length that was 40.1 to 44 millimeters, and the most common genus was Sternera. It's very hard to do accurate statistical analyses on things when you only have a couple members or one member representing your entire group. The example I wanted to bring up is hair color. If you wanted to look at the hair color of the UCEAP population, if you only looked at me for brown, well, if you wanted to look at the occurrence of brown hair and you only looked at me, the rate would be 100%. But if you expanded it to Naomi and I, then the rate would be 50% all of a sudden. This is just to emphasize that large populations better represent, that populations are better represented with a large sample size. So in conclusion, more research needs to be done. A couple suggestions that I have are to use a more evenly represented sample population for the reasons that I just said, and also to look into why these factors were found to be statistically significant. Is it forearm length? Is it coevolution with specific genera? I hope that a future student can find it out for me. And lastly, I just wanted to do a big thank you. I know we're not supposed to have a long list, but I had a lot of people help me, so brace yourselves. The Estación Biológica, Estación San Gerardo, the Stuckey Family Farm, the Instituto de Monteverde, and Life Monteverde for allowing me to conduct this study on their land. I want to thank Emmy for help with ectoparasite identification. I want to thank the Fennel family for eating dinner with me at 10 p.m. every night when I got back. I want to thank all of the bat people, Maya, Steph, Aileen, Jackson, and the groupies, which were Nadia and Stephanie, for their help. Some of the best nights I had in the program were out there catching bats with you guys. And then last but certainly not least, Fede and Adri, your support was immense, and I'm so grateful that I got to do this. To everyone that I mentioned and to Costa Rica, you touched my heart. I'll never forget. Thank you.
Time for questions.
Okay, just a quick correction from what I thought you did great. You really pointed out the other aspects of looking at these ectoparasites, because after all these years, and I've been here, there have been very few studies of ectoparasites. And that's true in general, that there's not much. But I just want to make a little correction. You said by the beginning that 5% of bats had rabies. Well, no percent of bats have rabies, because rabies is a fatal disease. If a bat gets rabies, he dies the same way you would if you got bit by something rabid and didn't get the vaccine, you'd die. So there's not a bunch of rabid bats walking around out there. And most bats don't even carry rabies. Vampire bats can carry rabies, and the reason that people hate them is because they carry rabies to cows in some situations. So anyhow, just following up on the story that you've been hearing all along, the bats are the good guys. They don't go around. The chance of getting rabies from a bat are so low that I don't worry about it. I've been handling bats off with my bare hands for 45 years. I haven't got any yet.
Thank you so much. This is sort of the same thing I already asked in the last presentation, but I thought it was super interesting that one of your speculations was maybe that the forearm length kind of coincided with genera, because that's might be true. But did you do any research into the brooding behaviors of the most abundant, the group with most abundant amount of ectoparasites in relation to other groups that you studied?
Yeah, so that was actually a factor that I wanted to include in my study, and I was asking Andrea about, like, do certain bats have more ectoparasites because of their roosting preferences, and I tried to do research on it, but a lot of these species, they don't even know where they roost to begin with, so it's really hard to do a comparative study when, like, some of them don't have any data.
Right, like, it'd be super cool to do in the future.
I hope so.
Muchas gracias, Mari. Remember, I guess we're doing wait, what? Thank you, friend. Sometimes when you are in native bats, there is a bat flying around and it's not coming to the nest, and then little Frank is like a bat. Remember, people, that the questions and comments that you listen during these presentations are useful, and you can take advantage of that for your paper. Bueno, next speaker in this section is Yailin Santillán from UC Santa Barbara. She will speak about food selection behavior and echolocation analysis of captive frugivorous bats.
Analysis of captive frugivorous bats. So to begin, I'm very grateful that I'm here in Costa Rica because I have just seen a lot, and it's been quite a journey to say the least. But I've been informed on a lot of things, and I love to see the biodiversity here, and more specifically the animal behaviors in the biodiversity. Foraging to be more specific. And so what is foraging? Foraging is the act of an organism seeking food or provision. So the most modern example that I can think of is us going shopping and looking for clothes, or looking for a restaurant to eat at with our friends. So with this, I realized that foraging and behavior have been linked quite often in multiple studies. And so one place that I observed is like foraging behaviors is at the bat jungle that is down the street next to Stella's. And it is a bat sanctuary that contains phyllostomatidae bats, which are frugivorous or fruit-eating bats. And they are very important to our environment because, as Maddie said, they are pollinators and seed dispersers, which means that they keep the biodiversity alive and overall are just very important to us. So at the bat jungle, the behaviors that I observed are hovering, which is they would just observe and fly over the plant for a second or longer. Circling, which is they would take a lap around the plant that I displayed, or they would pass by, take a note of the fruit, and then return back, and that was classified as circling. Landing is they would land onto the fruit, and either whether or not they would eat it or not, it would still be categorized as landing. And then the last one is biting, which is they would go up to the fruit, bite it, and then retreat very quickly. So with these behaviors, they could be due to a variety of things. Researchers have studied the behaviors of bats throughout quite a bit, and olfaction, which is the sensory cues are used. Visuals has been up for debate, but more recently has been said that they are very important for looking for food, and then as well as echolocation, which I think is a really, really cool mechanism that bats and other mammals like dolphins have, where they emit sound frequencies to an object. That sound frequency bounces off that object, and then the bats receive that frequency back, and that helps them determine where the object is at and the 3D configuration of that object. So with these behaviors, it made me question, since most of the research done on this has been done in bats in the wild, I was wondering about the bats in captivity. So then it led me to my questions. What behaviors do frugivorous bats display in captivities between fruits, and is there a difference in echolocation emitted by the bats between the fruits? So for my methods, I conducted this at the bat jungle in the enclosure that they have. And I began by creating 3 experiments, and in those 3 experiments, I did 3 trials. For all the experiments, I took down the feeders that they put up so that it wouldn't interfere with my fruits, and each fruit had was set up for 20 minutes. So then with that, I set up the fruit. The fruit would be set up right here, and here is a steer and home camera, so I could record and I could see the behaviors that were being displayed. And then here is a phone on the phone holder connected to the Pearson acoustic device so that I can later download and look at the echolocation calls. So for the first experiment, I did the fruit preferences between bananas, mango, and watermelon. The second experiment was between unripe and ripe fruit, so it would be ripe banana and mango and unripe banana and mango. And then the third experiment is fruits, unfamiliar fruits, which are fruits that are found out in nature that they're not used to anymore since they're in captivity, and familiar fruits, which is the feeders that they have diced up fruits, and that is prepared for them. So for my first experiment, I counted over 1,000 visitations for watermelon, which is the most visited fruit that was for the first experiment, and they displayed mostly a biting behavior. And then the second one was banana, which also displayed mostly biting behavior, and then the last was mango, which they displayed a hovering behavior. And then, so this is, the x-axis is foraging behavior, and the y-axis is the total number of visitations, and then the graph to the right is the same thing as this graph, but just in percentages. So I know this is a lot, and I don't mean to scare you with all this, but I really wanted to emphasize and show the importance of the echolocation calls between all of them, and I felt like if going back and forth on slides, you'd get lost. But all along with these echolocation calls, the visuals, they all are different between the fruits, so the land up, or the hovering up on the far left corner is different from the hovering on this fruit, which is the drawn to scale fruit. And then the hovering on the bottom corner right here is different from those two as well. So there's no, the echolocation calls are different in this experiment. for the second experiment in resulting the ripe banana and the ripes and the unripes, I used the same data from the first experiment on the ripe fruit, because the banana and the mango were ripe in the first experiment, and then in the and for the unripe banana and unripe mango, they tended to be visited less compared to the ripe fruit, but overall the ripe banana was visited the most. And even comparing the unripe, the unripe banana was visited the most compared to the unripe mango. And the same thing can be said about the echolocation calls emitted by the bats. The numbers are just very different, and for the mango, I did not record any bite or landing in the 3 trials that were conducted. So for the last experiment, comparing the unfamiliar fruits, so the fruits on branches, to the prepared fruits, the most visited was the prepared fruit, so it was the fruit that they are given every day in captivity, and to the branched fruits. So the prepared fruit had displayed most of a biting behavior, and then the branched fruits displayed more of a hovering behavior. And so I had initially thought that there was a similarity, not between the fruits, but between the behaviors of fruits. So for the wild figs, the hovering and biting behavior had similar minimums, and so did the wild guava. And in the echolocation calls, the hovering and the biting seemed fairly similar, so I thought. So in conclusion, bat preferences in fruits is that they like the easily accessible ones, and the ones that they visited the most, they displayed biting behavior overall, which is, it's concerning that they prefer the easily accessible ones, because if they were to be released or somehow escape, easily accessible fruits that are diced up and cut up for them already are not found out there, and they could probably starve if they do not focus on finding other types of fruits. But the good thing is that the fruits that they didn't like the most, or didn't visit the most, they still hovered over them, which is, previous studies has declared that that's sort of a way to inspect the fruit quality, and that means that they're still curious enough to test out the fruit, and that encouraging behavior that they haven't lost. So with this, I recommend that sanctuaries and conservations, they should display the food to the animals that they have, like they would see in nature, the best they can, so they wouldn't forget these foraging behaviors, because I believe that these foraging behaviors are very important to animals, just in case they do somehow end up back in the wild, or if their purpose is to go back in the wild after conservation and sanctuaries. But as for the acoustic portion, it has not been studied a lot, so it has left me with more questions, because what I initially thought before, upon further investigation on the last experiment, is not what I had thought, and so yeah, I'm open to questions or comments now regarding anything about acoustics or my overall presentation, as I am now finished. But firstly, I would like to thank everybody who helped me with this study, and I couldn't have done it without you. And yeah.
So you did your study experiment out of that jungle?
Yes.
Is there are they all from the wild? What's the population like? Are any of them born there? And you just have never seen in the wild, or are you rescued?
There are some that, from what I've heard and learned from them, was that they do get them from in Monterey, like the ones they find around this area, and then some of them, they don't breed in captivity, but they
species that breed very readily in captivity. We were afraid we'd have a population explosion in initial opening years of that jungle. And along came the government and said, well, you're not allowed to breed these animals in captivity. They might escape use. The objections were ridiculous because all the animals were from Monteverde. They're animals that fly around right with children looking. But in any case, we're not allowed to let them breed. And yes, occasionally we're allowed to replace animals that have died with new ones that we may get pregnant females with. So normally there's our population tends to gradually diminish, and then we either ignore the government and go out and catch some more, or we go by their rules and go through all the paperwork you have to go through to get permission to catch up. So it's a pain to have a zoo here.
Okay, any other question?
Yes.
Last question?
Was there any correlation between time of day and feeding experiments? Did you try to, make it consistent?
Yes, so I did the experiments between 10 a.m. and 1 to 2 p.m. So their feeding times are around 8:30 a.m., 12:30 p.m., and then right before they close and like go to sleep. They're on a flipped day and night schedule.
Right.
So like around 3 p.m. But that's generally they feed throughout the day. So I just did it within those times because those were the more realistic times that I was able to be there and record and set up everything and do it as much as I could.
That's your thing.
Alrighty, hello everyone, and welcome to my presentation on the effects of land use on diet richness of Monteverde's nectar-averse bats. So to begin, pollination is an imperative step in angiosperm life cycles all around the world. More than 80% of plants in Costa Rica and the neotropics are pollinated by animals such as hummingbirds, bees, insects, and bats. And these are crucial for reproductive cycles of plants. And I studied bats within the Glossophaceae subfamily within the Philostomidae family because they're a model of pollinators in the Monteverde region. And they're crucial for both export crops such as bananas and coffee, as well as native ecosystems that we find here in Monteverde, such as pre-montane wet forests all the way up to cloud forests. However, land use and human development in the region is very impactful on ecosystems here and has many implications. So, for example, ecosystems that are undeveloped, such as cloud forests, This ranges all the way up from cloud forests to formerly developed ecosystems, such as reforestation efforts, and currently developed regions such as pastures and farms. Land use affects all pollinators and affects their access to different plant species, and this leads to a loss of diversity with heavy human impact. And to understand the trends of my study, I interpreted pollen diversity found on pollinators as an indicator of the plants that they visit, and the diversity of plants visited as the diet richness of the pollinator. So after researching the indicators and varying land usage here in Monteverde, I asked the question, is there a difference in the diet richness of nectarivorous bats across varying land use in Monteverde? And I hypothesized that because plant diversity is greater in more intact ecosystems, there will be an inverse relationship between land usage intensity and the pollen diversity found on these bats. So I chose to study 3 sites of varying land use intensity within Monteverde region. Starting in the undeveloped, this is the Estación Geológica where we stay. So that is surrounded by undeveloped intact cloud forest. The next site is the formerly developed Institute Trails right in front of you guys this way. That is a reforestation effort about 25 years old, so it's primary growth forest. And final site is Life Monteverde, which has been cultivated for coffee and surrounded by pastures for several decades. For experimental design, I surveyed these 3 sites over 6 nights between November 11th and 23rd using mist nets to capture the bats in the evenings. And we measured species, mass, forearm length, pollen presence, and sex. And for pollen sampling, I used clear adhesive tape to dab off any pollen found on the nectar bats that we captured, and then placed them on labeled slides to later examine within the lab. Here we are setting up the nets at the Estación and the processing station at Life. Some bags with captured bats ready to be processed, and some aspects of processing, such as the forearm with calipers. These are some bats that we caught. We have an insect bat, a fruit bat, and a nectar bat, for example. And finally, after the nights in the field, I studied pollen samples under a microscope to identify pollen morphotypes, and those are examples of the slides. And I used pollen databases and previous research to determine the lowest taxonomic classification for the pollen that I was able to find. So in total, I caught 8 nectar bats and found a total of 23 pollen morphotypes. And just a reminder, my hypothesis said that less intense land usage would mean greater bat diet richness. So here are pictures of the 23 pollen morphotypes that I was able to identify. I have found 5 morphotypes at family level and 6 at genus level, and 11 went unidentified. So to begin, the total number of pollen morphotypes at each site in increasing intensity. Site 1 had 16 at 3 sampled bats, Site 2 had 10 at 1 sampled bat, and Site 3 had 11 pollen morphotypes among 4 bats. Now for mean or average number of pollen morphotypes per site, Site 1 had 8.7, Site 2 had 10, and Site 3 had an average of 6 among 4 bat samples. And because the single sample size of Site 2 throws off averages and total numbers, it's better, in my opinion, to examine the trends using the 2 extremes, which had both the same species of bats as well as more than 1 bat sample in each site. So this is comparing Site 1, the undeveloped, versus Site 3, the currently developed. And we can see that not only is the total and mean number of morphotypes greater at the Estación, but the number of unique morphotypes and the percentage of morphotypes unique to each site is greater at the Estación. So this leads me to conclude that human development and land usage negatively affects the diet diversity or diet richness of these esophagean bats. And beyond this relationship between land use intensity and pollinator diet, other factors that can affect this apparent trend involve sample size. Like I said, I only found 1 bat and 8 at 1 site and 8 bats in total. So larger sample sizes would allow for more accurate numbers. feeding range. In this diagram, for example, a bat feeding at site 1 would not necessarily be restricted to feeding only there. They may be able to move beyond this region to feed, which could throw off the pollen count. Bat species as well. I found 7 Glossophatina soricina and 1 Anura jeffreyi, and those have 2 different jaw morphologies, which allow them different access to different flowers. So that could be showing different pollen morphotypes in different samples. And finally, pollen consumption. These bats need to supplement their diets with protein, and one way they do that is by licking themselves clean of the pollen that's on their fur, and that could be restricting the amount of pollen that I find in each sample. And future research would be helpful in understanding larger sample sizes. So if we found larger values of samples at each site, that would create more accurate numbers, as well as researching the same hypothesis with different species, such as hummingbirds or bees, for example, which could model this trend in land use versus diet richness, based on the pollen effect. And in conclusion, understanding pollinator diets allows us to interpret the impacts of human development in important ecosystems like the Monteverde region. Pollinator diet richness is important for agricultural resilience in how pollination is important for crop cultivation and the continuation of Costa Rica's export crops, like I mentioned, bananas, coffee, pineapple, things like this. Ecosystem diversity also relies on pollinator health. Many groups of pollinators facilitate the life cycles, like I said, of plants in the area, so they are imperative to the survival of these species. Costa Rica and their ecotourism economy rely heavily on the natural beauty of the country, and that's only held up through the pollination by these animals. And finally, reforestation efforts like the one we have here at the Institute are only made possible by species like these bats and like these insects and hummingbirds that allow for plants to spread and grow over time. And finally, I'd like to acknowledge my advisors, Fede and Adriana, as well as the staff and faculty of the 3 sites that we worked at, as well as my bat crew and my homestay family. I appreciate everyone's help. It would not have been possible without you. And thank you. Any questions?
Very good use of the time. Opening for questions.
Right.
At any point, did you consider catching bats at the Hummingbird Gallery?
I did. There were previous student research that did use both, I think, your backyard and the pollen place, but I chose these 3 sites because the Estacion was a good spot to start, and then we went to Life for a few nights, and then we had originally tried the Stuckey farm, but it was a full moon and very windy, so last minute we decided to go here on Thanksgiving, so it was just a last minute location choice.
Were the numbers of measurements, were the numbers of times that you visited each site the same, and it just happened that there was only 1 bat caught here?
So this was the only site that we visited once. It was the last day of research, so we came here one time, and we only caught 1 bat. But we spent 3 nights at Life and 2 nights at the Estacion. It just, we intended on spending the same amount of time catching bats at each site. Some nights were cut short early because of rain or other factors. So time was our most consistent factor, except for this location. Right.
Kind of a silly question, but were you ever worried that like a tape was gonna like stick to the bat and
They have very fine fur, and this allowed me to just, like, I was able to wrap the tape around my finger and then kind of just dab their face. And we also had a UV light that I was able to identify pollen with, so I could dab my cat's faces. It was very simple.
Okay.
Gracias, Jackson. most interesting section. I would like to thank all the back crew for the opportunity to go to the midnets and grab bats again. And next speaker that I am pretty sure in the future will win an Oscar award or something for her actoral facilidad. Stephanie Diaz Martinez from UC Berkeley. She will speak about differences in tooth morphology between Sturnera mordax and Sturnera ondulensis.
Thank you, Billy.
Complicado, frankly.
Thank you so much, Billy. My name is Stephanie Diaz. I'm representing UC Berkeley and Bears. So today I will be discussing about the differences in tooth morphology between Sturnera mordax and Sturnera ondulensis. So let's begin. So Sturnera species are one of the most common genus in Costa Rica, as they are frugivorous bats that are found in middle to high elevations. With group variation comes differences in tooth morphology. I'm going to use this real quick. So both the Sturnera species, ondulensis and mordax, have the same dental formula, which is 38 teeth. So what dental formula represents is basically the number of incisors that we have, whether those are central incisors, lateral incisors, canines, premolars, and molars. So right here I have two images from the newest K guide that Richard Laval was actually a part of. And right here in this bottom image shows the mandibular incisors for both Sturnera mordax and Sturnera ondulensis, and they demonstrate how these mandibular incisors are bilobed for both species. Up here in this section A, I'm talking about Sturnera mordax. Their central incisors are bilobed blunt with converging tips, whereas for Sturnera ondulensis, their central incisors are unicuspid with diverging tips. So these are for the maxillary incisors, by the way. So tooth characteristics is one of the determining factors in distinguishing ondulensis with mordax, since both species have dark brown, medium to dark brown bodies with reddish patches on their shoulders. It also allows us to figure out their choices and behavior. So these creatures have displayed land restoration along with seed dispersal, and their teeth variations have something to do with it. Unfortunately, looking deeper into the differences of these two species, along with comparing their teeth sizes, there seems to be some critical factors that have been understudied. So this leads me to my question. How does tooth morphology between Sturnera mordax and Sturnera ondulensis vary in Monteverde and in San Gerardo? So I conducted my research in mid-November at the Monteverde Estación Biológica at the Life Farm, the Monteverde Institute, and the Children's Eternal Rainforest. So for each site, we set 3 different mist nets in different areas, and once the evening began, we checked the nets every 15 to 20 minutes until we started our bat processing. So once that had occurred, we got the bats, we removed them from the mist nets, we measured the weight of the bat inside the bag, and then we subtracted the mass of the bag later. We measured the forearm length with a caliper, and then in order to determine the age, we put a light underneath one of the bones of bat wings, as mentioned before by one of my colleagues. So if the light fused, meaning if the light was fused when we put the light on the bone wing, it would be considered an adult. But if the light went straight through, it would be considered a juvenile, as its bones aren't fully developed. Next, we would determine the sex, and then along with that, I would take images of the incisors
I would take these images, import them into image J, and because these bats are quite feisty, it was kind of difficult to put in a ruler as I was taking an image with the magnifying lens. So what I did in image J was get the diameter of each magnifying lens in each image, and that's how I would set my scale. And from there, I would measure the width and length of, both central incisors, mandibular and maxillary. So, my results. I captured a total of 36 species. The sample sizes, it's important to clarify, for Hondurensis, Mordax, and Parvidens were 32, 2, and 2. And I think that is important to keep in mind because if I probably collected more Mordax, maybe these results would have looked differently. I incorporated Parvidens on here, and this is for mean mandibular length along with the mean mandibular width. And then in these next slides, I have the mean maxillary length along with mean maxillary width and the mean forearm length as well. I incorporated these results with Parvidens because you can see that for most of them, they're quite different compared to Mordax and Hondurensis. And that is because those are quite easy to identify since they have trilobe mandibular incisors. So really, the study here is with Mordax and Hondurensis, but I couldn't leave out the Parvidens. But we can see some similarities in the results, which makes sense because they're two different species, but at the same time, they're pretty close. So I decided to run some t-tests for all of these bar graphs, and the results showed no significant difference. So what is the reason for that? Well, these two bats come from the same family and genus, so it makes sense as to why they're almost identical. But it is important to mention that during one of our nights bat processing, I encountered some bats that had forearm lengths of a Mordax, but tooth morphologies of an Hondurensis. So I'm thinking of two ideas. One of them being tooth attrition. So it might play a role here as these teeth wear out over time, diminishing one of their identified factors, which would be the lobes of the incisors. I was recently asked as well, wouldn't this apply to both species? Yes, it would, but it would be more prominent in the Sternio-Mordax because their maxillary bilobed incisors are there, whereas for Hondurensis, it's just unicuspid. So maybe next time we can study the symmetry of the central incisors to see if teeth attrition is a legitimate factor. But bat hybridization could be another idea. What if these bats mated with each other? Sheep can breed with goats, horses can breed with donkeys. If interbreeding happens with other groups of mammals, why wouldn't it happen with Chiroptera? So ultimately, in this study, there wasn't much variation in tooth morphology when it came to measuring sizes of length and width. Again, it could also be because I didn't capture that many Mordaxes. But to have a more definitive understanding of the variation between tooth morphology, I do recommend future research to be conducted. This study would reveal the evolutionary, molecular, and ecological significance of bat hybridization and dental morphology in Sternio-Mordax and Sternio-Hondurensis. Acknowledgements, I didn't incorporate other things that I will talk about right now. I want to give thanks to Federico and Adriana for devoting so many nights. I also want to thank my bat crew, and I want to thank all of the areas and stations in which I conducted my research: Life Farm, the Estación de Monteverde Institute, Children's Eternal Rainforest. I also want to give a huge thanks to my host family. And Richard, thank you for creating Murciélagos de Costa Rica book. I love it. Thank you.
us about any difficulties you had in getting them to smile for the picture?
Yeah, well, I mean, besides almost getting bit, I think it was challenging to get the correct lighting and to get a good quality of an image, because I wanted to make sure that I was getting, you know, all the way to the root, the periodontal area of the incisors, because if I don't do that, then I'm not really measuring the exact length, right? So, yeah. Yes.
Did you use methods like imprinting, like letting them bite onto a piece of foam or cardboard?
Yes, as a matter of fact, initially I also wanted to incorporate, you know, their bite and kind of make figure out to see if there was any differences with Esterneria mordax and Hondurensis with their bites along with their tooth morphology. But the only resources that I had were clay. And if you don't move clay over time, it's going to become hard. Also, these are fruit bats, so they can smell the clay. But maybe next time, if I do have dental wax base, then I will do that.
When the bats saw Stephanie trying that, they started laughing, and this is where she took the picture.
Yeah, that is exactly what happened. Any other questions? Thank you.
Frank, do we have a break? No? Okay, as Frank said, it's not my fault, so next session is going to start.
Oh, wait.
We want a break.
Yes! Just
Take a break. Let's have 10 minutes of break.
Yeah, let's go do it.
about behavioral ecology of vertebrates. Our first speaker is Rich Lee, and she's going to talk about vocalizations of grass.
Hi everyone, I'm Trish, and the focus of my research is frog vocalizations in acoustically disturbed environments and the relationship of sound and tree height in proximity to water. So first of all, an acoustic community is comprised of all the organisms in a given area that produce sound, and each species occupies its own acoustic niche that is defined by their frequency range and their acoustic vocalization. And within a given environment, there are three common types of sounds to hear. One is biophony, which is sounds of animals, and geophony, which are sounds of the environment, such as wind and water, and anthrophony, which are human sounds. So why frogs? Well, frogs depend heavily on their ability to vocalize and communicate with each other through sound in order to survive. And they also tend to congregate around freshwater bodies, and these areas can be considered rich acoustic communities. But they also pose an issue because of the sound of water that can make it harder for frog sounds to travel. So this is also known as sound degradation. And this is why it's important to understand how background noise can affect frogs and how, in turn, they respond to these background noises. And this brings me to the acoustic adaptation hypothesis, or the AAH, and this states that animals will adjust the usage and the structure of their vocalizations in order to avoid sound degradation. And one method to record sound is through audio moths. They look like that. They're full spectrum acoustic recorders. And once you have these recordings, you can also use the acoustic indexes available through the Kaleidoscope Pro program to quantify the sounds of your recordings. And for my study, I used the Acoustic Complexity Index, or the ACI, to quantify all of the sounds within the environment. And I also used the Normalized Difference Soundscape Index, or the NDSI, in order to find a ratio of geophony to biophony. So the NDSI is usually meant for anthrophony and biophony, but since the sound of water falls within the same frequency range as human sounds, we are able to use this index for that as well. So my question is, how do frog vocalizations change from the ground to the tree canopy in response to sound degradation? And specifically, I'm going to look at tree frogs because they have the ability to climb trees and get away from the sound of water near the surface. And I picked two sites in the forest surrounding the Estación Biológica. Site A is a stream, and site B is a waterfall. And within each site, I also had two heights, one at 6 meters above the ground and one at 1.5 meters above the ground. So I'm comparing heights as well as locations with different amounts of background noise. In order to do this, I got this 6-meter pole, and I attached two audio moths to it, one at each height that I mentioned. And theoretically, this would show us where the animals are vocalizing from. And I configured the audio moths to record 1-minute recordings every 30 minutes over the course of 10 days and overnight from 7:30 p.m. to 5 a.m. So there were 5 recording nights per site, and in total I had 400 minutes of recordings, which is around 6.5 hours of audio data. So now on to results and discussion. This is a graph of the ACI analysis, and SH is the stream high, SL is stream low, WH is waterfall high, and WL is waterfall low. And basically this is showing that the acoustic information between treatments was similar, so there was no statistical differences, and you can tell when you look at these boxes that the averages are these lines that fall directly through them, and they're very close to each other. And this is basically saying that the acoustic community of each treatment was very similar. For the NDSI analysis, this shows that the acoustic information differed significantly among all 4 treatments, except for the last two, which is the waterfall high and waterfall low. And so which treatment has a higher ratio of geophony to biophony? So for the NDSI, a score closer to 1 indicates that there's more animal sounds than background sound, and a score closer to -1 is the opposite. So all of these actually have a lot more animal sounds than background noise. And this is kind of the opposite of what you would expect with the acoustic adaptation hypothesis. So this shows that the waterfall location actually has more animal activity, as well as the low locations of both sites. So here and here, they're both higher than the high locations, and so they also have more animal activity. And in addition to these acoustic indexes, I also went through each recording and marked which ones had frog sounds. So for site A, 154 out of 200 recordings had frog sounds, and for site B, only 28 had frog sounds. And for the low height, 77 had frog sounds and 9 had frog sounds for site B. And for the high height, also 77 recordings had frog sounds and 19 recordings for site B. So this is saying kind of the opposite of what the acoustic index showed. This is basically showing that frogs actually prefer to vocalize at the stream over the waterfall, and within the heights for the waterfall, they prefer to be higher, so further away from the sound of the waterfall. So these results are inconsistent with the acoustic indexes, and I feel like this is because the NDSI is usually used for measuring human sounds, and it might not be as effective with identifying and quantifying geophony. There was a study that used the NDSI for a similar purpose as mine, and they had accurate results, so I think this just shows that it's not as consistent as it can be, and further development is needed for these acoustic index tools. But in conclusion, the NDSI showed that more animal acoustic activity occurred at the waterfall and at low heights, and this indicates that frogs prefer to vocalize near the water regardless of sound degradation. But according to the frog data, there are more vocalizations at the stream and at high heights, and this indicates that frogs prefer to vocalize away from water to avoid sound degradation. And this would support the AAH, and personally I would say that the frog data is more accurate just because I went through it and I listened to each recording and marked which ones had frogs, and with the indexes you can't really see what exactly they're identifying to be geophony or biophony. But overall, further research is needed on the NDSI, and also in order to gain a more comprehensive understanding of the acoustic behavior of frogs and this relationship to the AAH, I think there needs to be more studies with different sampling gradients, such as a horizontal gradient or an elevational gradient. And this is especially important as the climate crisis increases, because we're going to see more extreme weather events, and this can lead to extreme thunderstorms, creating a lot of background noise, and we don't know how this will affect frogs that are not as capable of adapting to higher background noise. And these are my acknowledgements, so thank you. All these people, Adri, Eladio, Fede, and my home state family, and also everyone here, and the program for all the opportunities they've given me.
Thank you, Trish. We have time for questions. Who has a question for Trish?
Why do you think the frogs prefer the stream, or there were more vocalizations by the stream than the waterfall?
So, well, you can imagine how loud a waterfall would be compared to a stream. They're both moving water, but the waterfall is definitely a lot louder, and when I listen to recordings, you can definitely hear the sound of the waterfall more than the sound of the stream. And I also just think the stream is more, has less disturbance, so it's you would want to congregate around that rather than a waterfall.
With a waterfall being such a loud thing in general, I was kind of surprised to hear that the biophonies at the waterfall out-competed sound sound-wise with the geophonies, but then also the lack of frog presence at that site in comparison to the stream. Do you have any idea what biophonies were interfering or overriding the geophony at the waterfall?
Yeah, I actually have another slide. But so there were 3 frog species that I heard, and Eladia helped me identify 2 of them. These 2 that we identified were only at the stream, and this mystery one, I don't even know if it's a frog, but I'm gonna say it's a frog for now, and that was only heard at the waterfall. And like, when you compare what their vocalizations look like, these 2 have very like short, little vocalizations that like happen like once in a while in the recording, whereas the other one, it's like consistent throughout the entire 1-minute recording. And that also happened in the higher frequency range, so possibly that is what's like changing the balance of biophony to geophony.
Okay. Did you count that as a frog in your?
I did.
Okay.
Yeah.
How difficult was it to isolate the sound of frogs with all the active noise? And also, how long did it take you to go through all of this?
It's actually really easy on the Kaleidoscope program because like it visualizes the sound for you. So the frog sounds like are very distinct, and they're like they look, or the intensity is stronger, so the color is different as well. And then you can also make it so you only hear a certain frequency range as you listen to the recording. So yeah, it was okay.
How long did it take?
Not long, you know. Rachel?
Do the frequency ranges of, like, for example, those 2 frogs differ?
Yeah. For this one, this is the emerald glass frog, by the way, which Jana found at the photo. This is the photo. This one has the highest frequency range from, I believe it's 6,000 to 7,500 hertz, and then this one is within the 4,000 hertz range. Yeah.
Thank you, Trish.
So, to begin, I want to talk about how problem-solving capabilities in animals are widely studied. We can see this in the Mexican brown jays with the log puzzle, which I took a lot of inspiration from. The capuchin monkeys and a tube food puzzle done in a zoo in Mississippi, where the capuchin monkey was easily able to access some grapes in a plastic tube puzzle.
the agoutis and the coconut puzzle, as well as the coatis and the food maze, which were both conducted by UCEAP students in past years, in which the agoutis were able to lift a coconut up and receive the food reward inside of it, as well as the coatis, which they were able to navigate a maze in order to get to a food reward. In my preliminary study or observation on the magpie jays in Guajinikil, I saw a magpie jay land on a closed bag of chips and start pecking at it vigorously in hopes to get to the chips inside. And that is where I decided to ask this question. Is this sort of behavior and problem-solving capability similar across all jays and even across mammals? So, to begin, I started with building the log. This log is one of the most important pieces in my research, as it has the 3 challenges that I made for the animals. The log is a Cecropian log, so it has a natural tube on the inside, and I will utilize that tube for one of the challenges that I will talk about next. So, the 3 challenges include the top challenge, the pipe challenge, and the latch challenge. So, these challenges showcase different levels of difficulty, which is important because it will also reflect in the problem-solving capabilities of the animals that solve them. The first level of difficulty is the top challenge. It is the least difficult, in which they will have to grab the bananas off the top of the log. No funny business, just food on the top. The second level is the pipe challenge. It's the medium level difficulty, in which they have to utilize either tools or their arms in order to reach inside the pipe and grab bananas. The third level of difficulty is the most difficult, the latch challenge, in which there is a latch on the top of the log right here, and they have to either lift it or somehow pull it in order to access the food inside the latch. Okay, so this is how I set it up. First, I set up the camera traps, and then I set the log down and made sure to put bananas in the 3 locations: the pipe, the tube, the top, and the pipe. The latch, excuse me. Okay, let's restart. Latch, tube, and top. 3 locations. And then I utilized the stick in order to stabilize the log because it is cylindrical, and cylindrical things tend to roll. And then I just sat and waited, and waited, and waited until an animal happened to cross upon the log and solve it, hopefully. So every time the animal saw the log, I would refill it, and the process would go through all over again. Before all of this, I made sure to put up bananas and all kinds of compost for the animals to be attracted to the location, so they know food is in this location. Okay, so I used 2 locations. My main one was in Monteverde in Frank's porch. I spent a lot of time there. And in San Gerardo near the station. It was just down the path that we came towards it. So this is what my results look like. Starting off with total physical interactions with the log. As you can see, there were 70 coatis that made physical interactions with the log. The rest are there. You just have to look closely. There's 2 agouti physical interactions, 2 capuchin monkeys, 3 opossums, 2 brown jays, and 2 rats. Keep in mind the brown jays were my first target, and only 2 showed up. Okay. So by physical interaction, I mean that they touched the log. They didn't. If they look at it and they're like, "Oh, there's food, but I don't want to touch it," I don't count that. I only count when they touch it. So that's for the physical interactions. For the animals' overall versus overall success, again, there is a pattern. And you see the coatis with 29, but there's opossums that also got to it. The 9 successes with 5 capuchins, 2 brown jays, and 2 agoutis. Now, I measured success by the number of times in which an individual successfully grabs a banana. For example, if there are 2 bananas in the pipe and 1 capuchin monkey grabs 1 banana from the pipe, that's 1 success. Another banana is grabbed. That's 2 successes. Moving on to the top. This is the first challenge, easiest challenge. We have 15 with coatis, 2 agoutis, 3 capuchin monkeys, 5 opossum successes, and 2 brown jay successes. And then the pipe, the second level of difficulty. We had 17 coati successes in which they reached their arm and grabbed some, 2 capuchin monkeys, and 4 opossums. Now let's look a little closer at this. This over here is the coati reaching in to the pipe. He's very focused to get that banana. The coati successfully took out 17 bananas. Now, in number 2, we have the opossum. He's right here. He only came out at night, so I didn't get to see him physically, but the camera trap did. And he got 4 bananas from the pipe. And then over here, you see this little capuchin monkey reaching his little hands into the pipe, and he was able to get 3 successful bananas. Now, as you can see, there's a lot of data, guys, but the coatis dominated. 20 successes in the latch challenge. They were the only animal that was willing and capable of opening the latch in my study. Yeah, first place goes to coati, second place goes to coati, and third place goes to coati. All right, now let's discuss a couple things. As you guys noticed, or maybe you didn't, let me point it out for you. Coatis were the most successful amongst all the animals, amongst the challenges. Capuchin monkeys, though, and brown jays are known widely for their problem-solving capabilities. So you would have expected something else to come out of it, and I will explain to you why this may have been the results that we got. So as you can see, there's a lot of agoutis here. None of them touched the log. And over here, you see a coati being like, is this safe? Is this not safe? Basically, to summarize what I just said, certain animals were more cautious of the log, while others were not as cautious. The coatis being the less cautious, or the neophilic, while the agoutis and brown jays were neophobic. Neo meaning new, and phobia, of course, is a fear. So they were afraid of new things. Only 2 out of all the agoutis that I watched made actual physical contact and were added to the data. Same with the brown jays. A whole lot of brown jays were heard, but only 1 band of brown jays actually interacted with the log. So this study showcases that there were animals willing, the most important part, and capable in solving the puzzle. The log puzzle did solve it, but it doesn't completely showcase the solving capabilities of the animals that were more cautious of the log. For example, the brown jays, as well as the capuchin monkeys even, and the agoutis. So thank you very much. I'd like to acknowledge, this is very important, Federico, as well as Jorge for helping me build the log. Jorge's over there. Give a round of applause for Jorge. Without their help, I wouldn't have been able to successfully build my log challenge. I also want to thank Fede for his patience. And I want to thank Frank and Katie so much for letting me utilize their porch as well as their house. It was very kind of you. And Katie for being my buddy to talk to when no animals were showing up. As well as Elsa for providing me with fruit compost and being a friend to talk to again when I was waiting for hours for the Jays to arrive. And that is the end of my presentation.
So cylindrical, and it just kind of threw me for a loop. When you think of a log, you think of things like jutting out, but no, it's like perfectly smooth. Do you think that could have been a deterrent? Because they're like, why is it so round and flush and smooth? Why would I approach that?
So I think one of the deterrents might not have been how cylindrical it was, or rather maybe how in the playing field of Frank's front porch, there's normally nothing there. So to have a log appear out of nowhere with food on it, it kind of seems like a trap.
So
right.
The perfect hole.
Yeah, actually, Fede helped me find that one. So yes.
Do the coatis have any food aggression behaviors? I'm curious if they
begin with, the agoutis, they were just like sniffing around. They were never going to touch the log to begin with. But they did like chase after them in a video that I took a while ago. The coatis are also a little aggressive to each other sometimes. The males, they were most of the time solitary. There wasn't another male close by. But when the horde came by with like the females as well as the juveniles, they were scratching, screaming at each other and everything. But for the most part, they all received the food and were able to complete the challenge.
A follow-up to that, how often was it just a solo male versus the group?
So I think for the most part, I had one particular coati come back every single time and solve it. In fact, I was refilling it right after he had solved it, and he was just staying in the corner watching. He was like, filled it up yet? And then right when I left, he came to solve it again. So the solo males definitely knew what was going on, so they showed up more often. The horde of coatis, whenever they passed by, they kind of just go for it, but they never came back just because of it.
And you didn't do any tracking of which male, which...
No, I didn't do any individual tracking.
Could you mention briefly how the coatis deal with the latch?
Okay, so let's go back to this. Well, this slide is pretty good. One of their favorite ways to open the latch was to utilize their hands. So in this one, you can see him opening his hands, opening the latch with his hand, as well as this one where he would lift the latch. In the beginning, they didn't know how to open it, so they would open it from this side with the screws. But then after a while, they figured out it's closer to the end, and then they would do that. Another method, very popular, is to use their snout to just stick it under the latch and then pop it open. So those are the two methods that I saw.
Thank you, Counselor.
Thank you.
Counselor is taking the lab to the U.S.
I'm trying to convince Frank to come here, but he doesn't.
Okay, so our next speaker is Nadia Gonzalez, who is going to talk about hummingbirds.
As Abri said, my name is Nadia, and my project was on covering behavior of hummingbirds while feeding at the Monteverde Hummingbird Gallery. So first, a little context on hummingbirds. That is a hummingbird. So they're found throughout the Americas, and they play a very important role as pollinators of the environment. And they feed on special flowers that cater to their long beaks and their tongues. So usually they're, as you can see in that photo, they're like tubular shaped. And they're also brightly colored, so you find them like in bright red, bright orange, so they can attract the hummingbirds. And they're also very agile flyers, so they can fly in any direction, including backwards and upside down. And they also do something where they hover in place. So what I was really interested in was how they hover in place while they're feeding, because hovering requires a lot of energy. And to compensate for that, they spend a lot of their time feeding from feeders. So that leads me to my question of, why do hummingbirds hover while feeding? So in order to do this, I came up with a list of 8 ideas for reasons for why they might hover while feeding. And then for each reason I came up with, or for each idea I came up with, I designed an experiment to test that. So the first idea that I wanted to test was perch availability. So in order to do this, I put up 2 feeders, one without perches and one with perches, as you can see by the flowers. And then I would observe them for about 3 hours, notating which hummingbird species came to the feeder and whether they were hovering or perching whenever they fed from the feeder. And then for the next factor that I decided to test, it was number of perches, and I did that by
having one control with just the 4 perches, and then another feeder I decided to add a stick on the bottom, and then that way a bird could perch here and a bird could perch on the other side, so it would add 2 more perches. And then I had one more where I added another stick diagonally the other way, and that would add another additional 2 perches. So there was one with 4 perches, one with 6 perches, and another with 8 perches. And then I also wanted to test flower proximity. So I did that by putting 2 feeders really close to each other, and then I attached a stick underneath to keep them in place. And the inner flowers would just be the flowers that were closer together, and the outer flowers were the ones that were further apart. And the idea behind this was that one reason why hummingbirds may hover would be so they could get from flower to flower faster. And then I also tested nectar availability. I did that by having one just regular feeder that would be like the unlimited feeder, and then another one where I covered like the holes where it connected to the main compartment with plasticina, and that way I would just refill like the little tubes every 15 minutes, and that would be the limited food feeder. And then I also tested familiarity by just comparing the data I took from the feeders I put up to feeders that had already been up by the gallery. And then I also tested sugar concentration by putting up 2 feeders, one where it was a higher concentration and the other one was what the normal concentration would be. And then size and wing loading, I did that by going one morning to misnet birds, and then with the help of instructors, we caught some hummingbirds, and then we processed them by weighing them. And also, I would take a picture like that, and then I'd put it into ImageJ, which some other people have already mentioned, and I'd use that to figure out wing area. And then to calculate wing loading, I would just take the weight over the wing area. So these are the species that came to my feeder. So there's the striped-tailed hummingbird, green-crowned brilliant, purple-throated mountain gem, violet saberwing, lesser violet ear, and the magenta-throated woodstar. So for my results, since I did have 8 experiments, I had a lot of data, and I'm not going to be showing all that data, or else we'd be here for a really long time. So instead, I'm only going to show the factors which I found to have a significant correlation. So beginning with perch availability. So this bar right here represents the feeder that I put up with perches, and this bar right here represents the feeder I put up without perches. And as you can see, well, okay, the y-axis represents the percent of hummingbird visits, where green is the percent of hummingbirds that were perching, and blue is the percent of hummingbirds that were hovering. And as you can see, there is less blue in this one and a lot more blue in this one, and that's because all of the birds that visited the one without perches was hovering, like no brainer. And I ran a chi-score test on all my data, and I found a significant correlation, meaning that perch availability does in fact play a role in the hovering behavior of hummingbirds. And the reasoning behind this was that, well, the reasoning I came up with was that the plants that hummingbirds typically feed from don't have perches for them, and as a result, they're required to hover in order to get the food that they need. And then the next factor that I found to be significant was number of perches. So this bar is the one for 4 perches, this one's for 6 perches, this one's for 8 perches. And as you can tell, like roughly as the number of perches increases, the amount of hovering decreases, which is the blue in the graphs. And I also found a significant correlation with this from a chi-score test. So the reasoning I came up with this was based off of the last experiment that I just showed. Hummingbirds do prefer to perch, and based off of that, if there are more perches, then they would be more likely to perch. And then the final factor that I found to be significant was feeder familiarity. So this bar right here represents the EAP feeders, or the feeders that I put up, and those were considered less familiarity. And then this represents the gallery feeder, which is much more familiarity since it's been up for a really long time. And I found that there was less hovering on the gallery feeders as opposed to the EAP feeders, meaning that they hovered a lot less when they were more familiar with the feeder. And I also found a significant correlation on this after running a chi-score test. And the reasoning I came up for this was that one reason why hummingbirds may hover would be for safety, because whenever they perch, it takes time whenever there's a threat for them to get up off of their perch and fly away, versus if they were hovering, then they could just fly away immediately. So if they're less familiar with the feeder or like any sort of setting, then they'd be less likely to perch. And this, so while running the feeder familiarity experiment, I realized that there was was a significant difference on how hummingbirds, like just specific species of hummingbirds behaved between the EAP feeders and the gallery feeders. So I decided to graph it out to see what happens. And I found that the hovering tendencies decreased for every single species that visited my feeders versus the gallery feeders. More specifically, this is the bars for the violet saberwing. Red represents the EAP feeder, and blue represents the gallery feeder. And it decreased by 71.9%, the amount of hovering. And then over here for the striped-tailed hummingbird, it decreased by 84.6%. So, improvements. Since I did conduct a study at the Monteverde Hummingbird Gallery, it was very much a tourist area. There were plenty of people always walking around, and they'd always want to come closer, take a picture of the hummingbirds, and so on. So that could have affected some areas of my experiment. And also, more consistency would be better, because halfway through running all my experiments, I had to move the location of my feeder because the violet saberwing decided to be really territorial. It did not let a single bird approach my feeder. So I decided to just move the location, or else I wasn't going to get data. And for future studies, some factors that would be interesting to look at would be interspecific relationships and location. So over here I put a picture of Simposyum biologica. It would be interesting to run the experiment there and see how hummingbirds that aren't used to feeders behave, since all the experiments I ran were on hummingbirds that are used to feeders. And for the factors tested, I could run more trials, such as for sugar concentration. I only tested one other concentration of sugar. So if I could test more, maybe I'll find some sort of significant correlation there. And also just observing this behavior on plants as opposed to feeders to see if the hummingbirds would behave any differently. So in conclusion, I found there to be 3 significant factors, those being perch availability, number of perches, and familiarity. Hummingbirds prefer to perch, but they are forced to hover because of the plants that they feed from. And in situations where they are provided a perch and they still choose to hover, this may be attributed to the fact that they are in an unfamiliar situation, and as a result, they are hovering in order to have a quick escape from any potential threats. And yeah, massive thanks to my advisors, Fede and Naomi, and thank you to Martin and Vicky from the Monteverde Hummingbird Gallery for letting me use their area and answering any questions I had, and Audrey and Maddie for coming 5:30 in the morning to the Hummingbird Gallery to help us net birds. And then also the Fennel family, my homestay family, for just welcoming me, and I love them so much. And then the hummingbirds for just letting me do experiments.
Did you see the violet saberwings acting territorial with the gallery feeders, or was it just your feeders? And if so, what does that mean for it being an unfamiliar item and it being territorial over it?
Okay, so to answer the first part of that, they would act very territorial with both of them, but the thing is, I put my feeders more out of the way. All the gallery feeders are kind of close to each other. I decided to put mine more in the trees, and because of that, it was much easier for them to be territorial over just 2 feeders that were right next to each other, as opposed to all the feeders in the gallery. So yeah, and then what was the second part again?
What do you think that means, since they tend to not be as comfortable around it because it's not familiar to them, but they're acting territorial instead?
I think, okay, I thought the whole them not being as comfortable would affect whether or not they perch or not. It could be anything from they're not familiar with the feeder, so they're not sure if perching on it will they'll be able to, or they'll be able to support them, and stuff like that. So yeah. More questions? Yeah.
Given that they have so much of a preference for perching when they can, I guess maybe one thing I would add to your follow-up list is I would wonder if, given 2 different feeders or 2 different flowers or whatever, whether a hummingbird would prefer to visit the one with a perch versus the one without. And along similar lines, do you know if there are any plants that hummingbirds visit that do have a place where they could perch?
I didn't really look into how hummingbirds behave, the plants that they actually feed from, because I figured it'd be easiest to run my experiments where I knew there were a lot of hummingbirds in the feeders, because then I'd get more data. But that would be very it's something very interesting to look into.
Time for one more question? Just for fun, what's your favorite? My favorite? Do you have a favorite?
Not that I really like the hummingbirds that were the most elusive, just because I didn't see them as often. So I'd be like the green hermit at the hummingbird gallery, or like I think there was one more, striped bell. I really like those because after like days of just seeing the same ones, and I'd see like a different one, I'd be like, oh my gosh, there's another species. But yeah.
Okay, thank you, Nadia. Okay, so our next speaker is Reina Shellman, who is going to talk about Pilates behavior.
Okay, so my name's Reina, and I did my project on how food types create differences in interest-specific and individualistic behavior within a Pilates band. So interest-specific means interaction of two individuals within the same species, and individualistic is kind of just like a solitary kind of behavior of a singular Pilates within a band. Okay, so my subject was the white-nosed coati, scientifically called the Nassau naruka. They're widely distributed in North, Central, and South America, and they're found in rainforest, dry forest, and cloud forest. I think many of you know, but if they're found solitary, they're males, and if they're found in groups, they're females and juveniles. So I picked this experiment because I hope to be a veterinarian, and I love mammals and behavior, and so seeing them every day at the station really made me interested in what they were doing and why they made such crazy noises and why were they on my roof and at 5 a.m., you know? So that led me to my central question, which is, does the interspecific and individualistic behavior differ based on type and distribution of food present? So I would go to the Estación Biológica Monteverde, climb up that hill, arrive at 8 a.m. with Zoe, and stay there until 5 p.m. So I would arrive at 8, and then they would come out for food, and then they would leave for most of the day, and then come back around 4 or 5 p.m. And so I had the option to stay or go all the way down the hill and then come back up. So Reina had an added bonus of taking a nap. So it was really conducive for my naps. I did this over 7 days, and I conducted 3 experiments on all of 4 trials. So my first one was bananas in 3 groups. So if you can see here, this is my unripe banana, which is this little pile here. My ripe banana, which is the middle one, and then my overripe banana, which is placed over here. And then you can see these quaddies are starting to walk towards it. And they're placed about 1 meter apart. Then I had a dog food dispersed where I take dog food and just like fling it across the ground to kind of mimic insects and other stuff on the leaf litter that might be eaten by quaddies. And then I did dog food piled in 3 groups similarly to my bananas, so about 1 meter apart, and probably like a 1/3 cup I would put in a pile. I recorded for about 3 minutes, and every time there were about 15 to 30 quaddies, which is pretty interesting because normally quaddies come in 15 to 20, but it makes sense because we were eating a lot and we had a lot of compost, and we would give it out to the quaddies. They're like, yeah, let me be there every single day. So moving on. I did a thing called focal sampling, which is where you first observe the quaddies by themselves, and you write a list of behaviors that you see. And then you create a table where you have all those behaviors on the top, and then you film. So within my 3-minute video, I found 1 quaddie that I could track for 1 minute, and I did that for all of them. And so on roughly all of my trials, I was able to get at least 20. On my dog food, I was able to get more, which will be explained later. And then I did, so that's focal sampling, but then I did a thing called scan sampling, which is where I take those same videos and watch it for a minute and try to observe every quaddy and what behaviors they do. So I just tally mark everything I saw, which was really necessary to see the quaddies I was unable to see for a minute. They would like come quickly and then leave. So this is a key for my focal sampling. So I can't write crazy descriptions in my graphs. And so what I did was I used 1 to 2 words, and I had a series of definitions I had for them. So I'm not going to say them all, but running would be like actively in motion, right? Or eating, consuming the food given in the experiment. Not like, sometimes they would eat the food and they go off and forage. That wouldn't count. Like, it'd have to be them eating the food I provided. So there's about to be a lot of graphs. But I wanted to include a lot of graphs because my whole experiment is based on observation. And so being able to look at a bunch of graphs and see the correlations, differences, I thought was really important. So we'll start here with my x-axis, which is my observations and different behaviors, which was shown in the key. And then my frequency of behavior would be on my y-axis. And then my legend over here, like the colors were the differences in time. So I would I'd have this crazy table and I would condense it so that it wouldn't be like a tally mark on every single section. So something that can be observed here is that kind of towards the beginning of my experiment, there's higher levels of behaviors. And similarly here, this is at the very beginning, eating, jumping, foraging, standing, sitting are all very, the frequency is much higher than the rest. Here it kind of starts flipping the other way, because you can see halfway to 3/4 of the way of a trial of my bananas in piles experiment. You can see that there starts to be more behavior then, and then finally for my last trial for my bananas in piles, you can see that the most frequent behavior happens at the end, and this can be explained because there's very low levels of actual quaddies eating. So that means that while a few are being really territorial, these don't happen to be adults usually, adult females, the juveniles and the rest of the adults were either being chased, or they were scavenging or foraging on their own, or grooming, doing whatever else, because it can get a little crazy and they don't want to mess with the territorial quaddies. So then we have the dispersed dog food. This is something you can observe is there are, first of all, less behaviors because all of the quaddies were able to access food. And so you can see that mostly they're foraging or eating. And then it also happens at the beginning between 0 and 10 seconds. Same with the second one, the third one as well, and the fourth. So overall, there are less behaviors being observed because most of the quaddies are able to go and get and forage and find their food. And then my last trial is piled dog food. So here it also follows the same pattern of at the very beginning, all the quaddies run and they're eating, they're foraging, they're standing or whatever, and that remains constant between all 4. But there are more, you can see, there are more observations of behaviors of being chased and other than the other dog food because, why? Because they're in piles and they're not as easily to access. Okay, so then I decided, I realized that the juveniles don't get any special treatment. Part of the reason why I wanted to carry out this experiment was to see if the juveniles get any other treatment besides just protection against predators. And this is showing that that's not the case. So in the bananas, there were barely any of the quaddies, the juvenile quaddies were able to get any food. This is the number of juveniles eating quaddies out of the total eating quaddies and the juvenile eating quaddies out of the total juvenile quaddies. So as you go lower down and the dog food ones here, there's a higher percentage of juvenile quaddies that access food. So this kind of demonstrates that there could be a hierarchy involved between the adult females and the juveniles. So here, then going back to the banana experiment, I made a table to show that every single trial I had, the ripe banana was the most desirable, and then the overripe, and then the underripe. And each experiment was placed into they had two variables. So the banana ripeness, which banana ripeness is eaten first and which banana ripeness is finished first. And it was the same trend all throughout. And that could be because, first of all, the underripe ones, they're bitter and they're hard to take off the peel from the fruit. And the overripe is kind of conversely, because they have that long snout and the teeth are like way back here. So trying to gnaw, and it's something that just smushes, like an overripe banana, is kind of hard to peel out from the fruit. So that could be another possible reasoning for this data. So, discussion. I found high levels of aggression in fruit experiment, and part of the reason why could be adult females versus juveniles. The adults are always going to get to the bananas first. There's never been a juvenile in any of my experiments. And then the juveniles kind of stay back, and that's when they're either they're grooming themselves, they're interacting, or they're just staying clear of the adults. And then there's scarcity of food. Obviously, if there's 3 piles, it's harder to get your nose in there and get the food. And so sometimes they would like grab it and then run away. But all in all, just the bananas made it really hard for any more than like 3 coatis to access food. And then there's hierarchy among spans, which is what I explained, that the adults don't prioritize the juvenile at all, and they kind of know their place. So between dog food experiments, there's more aggressive behavior in dog food piles. This would make sense because they're all clumped, and there's less of an equal opportunity to get to the food. But that being said, all the coatis that came from my experiment, they all were able to eat. Just there's more tendency for aggression in the ones in piles. And so before there's a preference towards ripe banana because they're easier to extract fruit from peel. And then potential future research could be investigate further banana preference and the hierarchical tendencies, because I just barely scratched the surface on that. So conclusion, coatis travel in groups solely for protection against potential aggressors or predators, as in adult females do not cater to juveniles. There's less behaviors during dispersed food because there's more access to the food and everybody's just focusing on on getting the food. Juvenile qualities all emerged and were able to find food in both dog food experiments, which goes to show that having that foraging, if you're searching for insects, is a lot easier than if there's a banana or guava tree, because I only saw the adult coatis going up the guava tree as well. So that's that. These are my acknowledgements. Okay. I would like to thank my primary advisor, Federico Chuchilla, for all his wisdom and time, my secondary advisor, Adriana Arias, for her feedback, and the Estación Biológica Monteverde for allowing me to use their property. Finally, I would like to thank my host family for all their support and for making me feel at home, and a special thanks to Vita for always packing me a lunch for me to take to the station. And also, I'd like to thank the coatis for their cooperation and time.
Cuando ella termine puede decir lo de la foto.
Okay, so our next speaker is Maya Gupta, and she's going to talk about coatis.
Okay, so I chose to do my project on Nassau and Eureka, more commonly known as white-nosed coatis, and how their vocalizations correlate with distinct behaviors. Out of the approximately 300 species within the Carnivora order, a feature unique to coatis is their social structure, which is comprised of solitary males and matrilineal bands, which are composed of female coatis and juvenile coatis, and sometimes even incorporate unrelated individuals that are subjected to more aggression. In order to facilitate such complex social dynamics, they utilize auditory communication, which is especially beneficial within forest habitats where visual identification signals can get easily obstructed. These auditory communication vocalizations can be categorized into two main categories, non-aggressive chirps and aggressive squawks, which are differentiated based on their frequency and harmonics. Frequencies are defined as a number of cycles per a unit of time, and harmonics are multiples of a frequency in waveform. This led me to my central question: how does the behavior of white-nosed coatis correlate to distinct acoustic vocalizations, and how do aggressive female squawks vary based on the directed target? In order to test this out, I went to the Estación Biológica Monteverde from November 13th to 27th, from 9 a.m. to 5 p.m. And I recorded the coatis whenever they came using my phone, as well as the linear PCM recorder and a powered directional condenser microphone in order to control the direction I picked up the acoustics from more concisely. I then went through all the videos and audio recordings and focused on vocalizations that had distinct behavioral associations. And I uploaded them to the Wildlife Acoustic Kaleidoscope software. After uploading it, I measured the duration of each vocalization, the number of nodes, and the duration of each node. And I defined a node as a group of harmonics occurring within the same time interval. I also measured the frequency maximum for the first harmonic, the frequency minimum for the first harmonic, the frequency maximum for the third harmonic, and the frequency minimum for the third harmonic. And on the x-axis, there is time in seconds, and on the y-axis, there is frequency in kilohertz. My results showed 6 unique vocalization types and their subsets, those being foraging chirps of male, female, and juvenile coatis, female aggressive squawks directed towards male, female, and juvenile coatis, hostile male grunts, female warning squawks, juvenile aggressive squawks, and juvenile retreat vocalizations. This sonogram depicts the foraging chirps, and this table encompasses my measurements for all of the sonograms. As you can see, these are for the male, female, and juvenile foraging vocalizations, and the table includes the total number of notes recorded, the maximum number of harmonics per node, the average duration per node, the average frequency maximum and minimum for the first harmonic, and since the male and females had 1 number of harmonics per node and the juveniles had 5, I also measured the highest and lowest frequency maximum for all the harmonics and the highest and lowest frequency minimum for all the harmonics. This is a sonogram of the female aggressive squawks. And this is a table encompassing all my measurements of all the sonograms. I did similar measurements, except all of these ones had 5 harmonics per node, so I just measured the average frequency maximum and minimum for the first and third harmonics. This shows the hostile male grunts, which only have 1 harmonic. Female warning squawks, which occurred when a hostile male was approaching. Juvenile aggressive squawks, juvenile retreat calls. Okay, and then for my results, I use a chi-squared test, which is a method of statistical analysis used to assess the independence between categories through comparing observed data with expected data. For the male versus female versus juvenile foraging chirps, I found significant differences between the duration, the frequency maximum and minimum for the first harmonic. And then for the aggressive female squawks directed at females, juveniles, and males, I found that the duration was not significantly different. However, the frequency maximum and minimum for the first harmonic was, and the frequency minimum for the third harmonic was also statistically significantly different. And then I found my results overall showed that there were 6 different unique vocalizations, and then for the female foraging chirps versus the juvenile foraging chirps, the female foraging chirps were of a significantly longer duration than the juvenile foraging chirps, and the female foraging chirps frequency maximum for the first harmonic was significantly higher than the juveniles, and the female and males both showed 1 harmonic, whereas the juveniles showed 5 harmonics. And the increased duration of the female chirps emphasizes a previous hypothesis in a paper by Morello and others that chirps are likely to function as a contact call because females have more use for this contact call since they're within the bands than males would. And then the differences between the frequency maximums and the harmonics in juveniles and females may aid in identifying one another by means of age. And then for the female aggressive squawks directed towards males, juveniles, and females, the males' frequency maximum for the first and third harmonic was higher, as well as the frequency minimum for the third harmonic, and the juveniles' frequency maximum for the first harmonic was significantly lower. This suggests that females change the frequency of their squawks when males are present, which may be a possible strategy to warn other quaddies within the band of a hostile presence. This also may be a technique that females use where they increase the frequency of their vocalization when males are present to help quaddies within the band distinguish their vocalization from lower frequency male vocalizations. The juvenile aggressive squawks had significantly higher frequencies than the female aggressive squawks, which is likely due to different vocal maturities in the quaddies, but also may be used to distinguish if a juvenile is being targeted by a hostile presence as opposed to a mature female. All in all, quaddies have complex social dynamics facilitated by a wide array of vocalizations and behaviors. This study could provide the groundwork for future studies focused on determining what factors of quaddy vocalizations aid in individual recognition, and what differences in duration and frequency suggest about these intricate social dynamics. Observing these interactions further could enhance our understanding of parallel behavioral patterns across species. The intricacy and individualism displayed within quaddy bands serves as a reminder that animals can have social dynamics just as unique and complex as ours. Thank you.
Why did you choose the first and third harmonics as your sounds?
Well, I chose the first one because it was the lowest one and it was going to be applied to all of them. And then I chose the third one because some of them didn't have up to 5, so I thought it was a good baseline for the maximum because some only went up to 3.
Questions for Maya?
Is it feasible to have a sense of the sex of the juveniles based on sound as well as not?
I wasn't able to tell that because the juveniles are generally like higher frequency and shorter duration, so it would be very minuscule changes, and I wasn't focused on that.
Do you think it's possible to recognize individuals by their sounds?
I do think that's possible, and that was another part of my experiment that I didn't get to do the data analysis for, because that I'm also very interested in, and I think playback studies would need to be done to further determine that, but I think that other research has suggested that, and my research also suggests that, but I would have to do further data analysis on the playback studies that I connected to be able to determine that further. But it is a very interesting thing. Yeah.
More questions for Maya? No? Thank you, Maya. So, this is the end of our session, so it's time for a break.
Hang on a sec.
No break.
You still want to take a photo?
Yes, please.
Okay, so if you could move out quickly onto the grass because Elaine would like to take a group photograph. Anyone who's not in a group, Silvio, folks, están bienvenidos a estar en la foto. Okay? Bienvenidos.
Yeah.
Oh, that's yours.
Hi everyone. My independent research project focused on discovering whether anti-glare properties in transparent glasswing butterflies had any effect on their predation frequencies. So, a little bit of introduction and background. Camouflage is a form of crypsis used by many organisms in the animal kingdom to blend into their surrounding environment to avoid predators, as well known. Butterflies in particular use camouflage as a primary defense mechanism to hide against predators, thereby enhancing their fitness. They accomplish this through a complex array of scales on their wings, and they're usually oriented in a way that's specific to a certain background, such as leaf litter or tree bark. However, there is a unique tribe of butterflies known as Achomione, who boast transparent wings, which is hypothetically the perfect crypsis in all backgrounds. So they're commonly known as glasswings, and they accomplish this through modified hair-like scales, which leaves the chitin membrane exposed, which is naturally transparent. These butterflies are unique because transparency is a relatively rare phenomenon in terrestrial organisms. This is largely due to the behavior of light. When light moves from a medium of a lower refractive index to one that is higher, in this case, air to living tissue, when it passes through, some light is actually reflected backwards, and that's noticeable. So in the context of like survival and natural selection, this would put glasswings at a major disadvantage because it would be an additional visual cue to predators such as birds. How glasswings overcome this is they've evolved this nanoscopic layer of waxy pillars known as protuberances that sort of act as a buffer between air and living tissue and essentially reduce the glare to nearly zero. So with all of that knowledge, I wanted to answer the question, do anti-reflective properties reduce the likelihood of detection of glasswing butterflies by predators? So this project required a lot of setting up before I could actually collect any data. The first step was to actually catch the butterflies myself, which I did with an insect net around the Simposyum. After collecting them, I would freeze them for 2 to 3 hours and then pin them to an insect spreading board to allow them to dry. And that was about overnight. Once they were dry, I could start the wax layer removal, which involved setting them up on the insect spreading board with a sheet of just printing paper underneath it. And I used a clean, dry finger to gently rub against the wings. And that was good enough to remove the wax layer. I couldn't verify that I removed it 100% because that would require an electron microscope because they're so small. But you could clearly see that there was a very big difference in the glare between the control and experimental groups. Then after I created those two groups, I could set them up around the Simposyum and alongside the road and conduct my trials. So I would set them up at 12 p.m. every day and allow them 24 hours to be predated. 12 p.m. I chose that because birds are typically least active in the afternoon, so they wouldn't be hunted while I was setting them up. And I did a total of 50 butterflies, 10 per day, 5 control, and 5 experimental. So here are some pictures. I know they're hard to see, but this was the insect spreading board after I had frozen them and was going to dry them. The middle picture is the process of removing the wax layer. You can't really see in this, but it has a lot more reflection on the wings after. And that is an example of how I pinned it to a branch relief outside and waited. These flowers are the Santa Lucias. They are the glasswing's favorite flower. I caught pretty much all of my butterflies on or around these flowers. And this is an example of the experimental and the control group side by side. The experimental's on the left, and the wings are a little bit wider, that's the glare, and the control is on the right. So this graph is consolidated, all of my results consolidated, and it compares the number of butterflies eaten between the control and experimental groups by day. I conducted my trials for 5 days, and as you can see based on the data, there's not really a clear trend. It actually seems to alternate between the control and experimental groups every other day. And you'll notice that on day 4, no experimental butterflies were eaten at all. And I would guess that that was due to the weather, because it was especially windy that day. And so out of the 50 butterflies, 24 were eaten, 13 control, and 11 experimental. And based on those numbers, there didn't seem to be a large difference. And to confirm that, I conducted a chi-square test, which you can see in the left corner here. And my p-value was 0.57, which I will touch on a little bit over here. But an indication of predation in my experiment was the complete absence of a butterfly, because there were a lot of other external factors, such as winds that would damage the butterflies, unfortunately. So I couldn't rely on just these damages as an indication of predation. And these would be examples of what a missing butterfly could be. So, like I said before, the attack frequencies were not different between the butterflies with and without anti-reflexive protuberances. And based on my statistical analysis, there was no association between the removal of the protuberances and an increase in attack frequency, because my p-value was much greater than 0.05. So my results were not exactly what I predicted, but there is an explanation for it. That's due to the foraging techniques used by birds. Previous studies have shown that many birds rely on UV patterns to locate their prey rather than other cues. And glasswing butterflies actually do have these UV patterns. They have white patches of scales at the apical ends of their wings, and that could have served as an explanation for why experimental and control groups were created at similar rates. Future studies might might explore how other functions of these protuberances layers, and such as like anti-wetness, wings tend to stick together without scales in rain and other wet conditions, so these might be more effective than typical scales. Or other studies might try to follow the predation route, like I did, and see what other factors might influence predation frequency, such as removing the UV patterns on the butterflies. One interesting thing that I found while I was reading about these things is that glasswing butterflies, while they have transparent wings, they also have aposematic colorations at the edges, which sounds kind of counterproductive, because if you have cryptid camouflage, the goal is to not be noticed. And so one of my predictions would be transparency is the primary defense, and if they are noticed, they could use aposematism as secondary defense. Yeah, that's it. Thank you to Emmy and Naomi, my advisors, for their guidance in this project. Thank you to the Simposyum for letting me use their area as a study site, and thank you for listening at this brief conference.
There's time for questions. Was there any, were there big differences in where you placed each of the butterflies that could have influenced whether or not they were predated upon, like in a thicker part of the tree was kind of covering it or not?
Definitely. Yeah, that was really difficult to standardize. I tried to at least put them in areas where they were exposed to sufficient sunlight so that they would actually reflect light. But that's definitely a factor that could have heavily influenced my results. Yeah.
Could you show the graph again? Like, I think it was the first bar graph, it was black and white. Yeah, that one. So the black is control and the gray are the experimental?
Yes.
And what's showing is the number eaten or the ones that were not eaten?
This is the number of butterflies eaten
So,
in each group.
and And these things are side by side.
No, the butterflies were placed not near each other, but like, I'd say at least like 20 meters away from each other. So they weren't like super close.
Because, I mean, on day 4, that's a pretty dramatic difference, right?
Yeah.
So, I mean, is there anything else that would help explain that or understand that? Like, maybe a predator learned about these guys? Or maybe the lighting was such that one particular butterfly wing was way more obvious on a certain condition, like a cloudy day or a super sunny day. I mean, again, that day 4 to me especially is a very striking result.
Yeah. I don't really have anything that I could use to explain that. I did try to also kind of randomize where I placed the butterflies so that predators wouldn't learn and come back to the same spots over and over again. But yeah, that was just an outlier that I wasn't expecting.
Besides birds, is there any other predator?
Yeah, sometimes other insects will prey on butterflies, such as spiders if they get caught in the webs. But in this case, the main predator was birds, and I believe that the birds have done it.
In the place where you put the butterflies, were places where you saw the butterflies before?
Yes.
The butterflies that you use, did you select them by being males, females, or again, or?
It was, I narrowed it down to a genus, Greta. They are like the truly transparent butterflies. So I stayed away from the more opaque ones and focused on the genus.
More questions? Okay, thank you, James.
No, es el adaptado.
Hello everyone. Today I'm going to talk to you about my project. It's about the acoustic communities of terrestrial invertebrates and their ability to adjust to anthropogenic noise pollution. So in order to get started, I have to define a couple terms. So I used bioacoustics and soundscape ecology to study the methods of their adjustments. And so let's just define those terms. So bioacoustics. Bioacoustics is the study of the production, transmission, and reception of animal noises, which includes human, insects, fish, all kinds of animals. So this study focuses on sounds from the order Orthoptera and the order Hemiptera, which is cicadas. So with that, sounds from this study are created from mostly males by stridulation, which is the rubbing together of two body parts, so like wings. Timbling, which is used by cicadas. It's like when they vibrate their exoskeleton, and then they use their large body chambers as a resonance chamber to make it really loud. So that's why cicadas are really loud. So with this study, I analyzed spectrograms, which just shows the spectrum of frequencies from audio recordings. So as you can see, this is an example of one. The y-axis is in kilohertz, and the x-axis is in time. So soundscape ecology. Soundscape ecology is the study of the acoustic relationships between living organisms and their environment. So that can be anything. That's in this case, it's between humans and insects in Orthoptera. And in this study that goes into soundscape ecology, I used 4 acoustic indices. First one being the NDSI, which just measures the difference between frequency bands of anthropogenic noises versus biological noises. And then acoustic community index from past presentations, you guys know what that is. ADI and AEI have inverse relationships to each other, so we'll get into that later. So there are 4 types of sounds, as Trish mentioned earlier. So there are geophonies, which is earth-made sound, wind, rain, waves. Anthropophonies, which are human sounds, cars, vehicles, stuff like that. Biophonies are made from animals. And technophonies, which are electromechanical noises, which we won't get into. But the first 3 are the main focus of this study. So that leads to my central question. How do acoustically communicating terrestrial invertebrates adjust to anthropogenic disturbances? So in order to go about this, I picked 2 locations to study. One is an anthropogenic site, right here. It's about 5 minutes south of downtown St. Helena, so I used that site as like a full anthropogenic site. It was near a road, so there's lots of noises going on to disturb the environment. And then a non-anthropogenic site right here, just off a trail near the Estación Biológica. So that served as like a non-disturbed site, completely isolated from the city. In order to collect audio, I used a sampling rate of 48 kilohertz in 16-bit folders, using a handheld recording device and an attachable microphone with a windbreaker. So I just held it up like this. And then I recorded 60 seconds every 9 minutes for a total of 12 minutes per session. And the session was one night. So for a total minutes of 60 minutes for audio files per location. And so to analyze this data, means of the data were calculated for each minute. So for example, I grouped up the 3 minutes from minute 1 and took the mean from that. And then I analyzed and calculated the acoustic indices using Kaleidoscope Pro software. And so here are the results for the NDSI. So as you can see, there was a huge difference. There was a significant difference between the station, which is the red graph here, and the Santa Elena, which is the non-anthropogenic site, and the orange graph here. So it was very different, very, very different. So in order to read NDSI, the closer the number is to 1, the more biophonies are in the area. So it just eliminates, so like if it's completely 1, there are no anthropophonies in the area. So yeah, this is just a box plot resembling the information given here. So this shows that the sites were correctly differentiated as anthropogenic and non-anthropogenic. And so here is a graph of my ACI. So as you see, sort of the orange here, on average there are more biophonies at the anthropogenic site than the non-anthropogenic site, which was a bit surprising to me. So this is just a box plot of that same graph from before, so it's the same. So the non-anthropogenic site had a much wider range of ACI, which could mean a lot. So this is the range of ACI at the anthropogenic site. So there was a lot of noise going on, that's what that indicates. And there was a lot less noises sometimes going on here. It just varied a lot more than the non-anthropogenic site. And so this is graph of the acoustic diversity index. So as you can see, there's a clear pattern between both sites. So as one went down, the other went down. This is completely separated too. So it's cool to see this pattern. And after about minute 5, the ADI at the stations kind of leveled out, but the Santa Elena ADI was much more variable, as we'll see, as we've seen before. And then the AEI, again, the AEI and ADI are inverse relationships, so it resembles about the same kind of pattern here. The non-anthropogenic site was more acoustically even than the anthropogenic site, and this graph actually demonstrates what the dust course is. So the reason why I took data from 5:30 p.m. to 7:30 p.m. every day was to take into account the dust course, which is just accumulation of a lot of insects and other animals, like calling together in order to isolate from predators. Discussion and conclusion. So this led to my question of how does a highly active anthropogenic area have a greater acoustic complexity than a non-anthropogenic area? I thought this was a very interesting result. And so after looking and analyzing these spectrograms of these audio recordings, I came up and reading some articles, I came up with the idea of acoustic masking, which just means that like insects can adjust their calls by increasing or widening their frequencies depending on their environment. So we'll see in the next couple slides how they did that by looking at the spectrograms. And then another thing they did was acoustic niche partitioning. So we'll see that like the more biophonies that are in the area, the more they'll have to compete with each other. So yeah, they want to minimize overlap with other calling individuals. And so this chart right here, we can just see like, this is how I analyze the spectrogram. So with the bandwidth, these are strong calls, and then these are weak calls. So when a call does not repeat, it is considered weak. And when a call is strong, it tends to repeat. And then in regards to dimensionality down here, unidimensional means just like one frequency. So at like 3 hertz, unidimensional. And so multidimensional means it's like at 3, 5, 7, like it goes higher and higher at the same part. So this is day 3 of minute 4 recording at the non-anthropogenic site. So you can see like there are a lot of multidimensional modulated calls ranging from 4 to 12. So it's a big wide range. And then this actually happened in all 30 recordings with 0 occurring at the anthropogenic site. So I thought that was very interesting. And then biophonies were found from 2 to 22 kilohertz range. So as you can see, there's a lot of modulating sounds coming from up here and a lot of multidimensional sounds here. And acoustic evenness was extremely high for the sites. And then day 1, minute 8 recording at the anthropogenic site. Acoustic masking occurred following the anthropophony right here. This is just a car moving by where I recorded at the left corner of the screen. And right after, you can see there was a call that started. Oh, I think it's there's a call that started right after that. So that's the niche partitioning. That's like it got quiet right after, so something started up again. And 90% of biophonies this site was found between 3 to 6 kilohertz range, which goes with the niche partitioning hypothesis. And yeah, I'd like to thank my primary advisor, Adriana, and the secondary advisor, Adelia, my host parents, Jenny and Wilbur Solis, and of course the Institute. Thank you.
There's time for like 1 or 2 questions.
Were you able to distinguish what kinds of what kinds of organisms were localizing at the anthropogenic site?
Well, it was mostly, I did catch a couple cicadas, but those were kind of rare. That was only on like 1 every like 5 recordings. But it was mostly like crickets and grasshoppers, like those. Yeah. Pushing back there.
Yeah. I'm just curious, where did you put the audio or your recording device at the Simposyum? Because I'm pretty sure it was windy at the time you were doing it, and that could be an explanation why you didn't hear.
Yeah. So with recording, with analyzing ACI takes into account geophonies too. So that's probably another reason why the ACI was so high for the anthropogenic site compared to the non-anthropogenic site. And so I had to take that into account when analyzing the results.
Thank you.
Yeah, is it doing the presentation here, or is it doing the...
It's so funny.
Okay, our third presenter is Matthew Cole, and he's going to teach us about this very common and very understudied fly, the present here in Montreal. Thank you.
Thank you. Okay, yes. Hi, I'm Matthew. Today I'd like to talk to you about my study testing the novel orientation ability of Stenomica flies. So orientation is sort of the ability to understand one's position in relation to like the space around it. So obviously that's incredibly important for navigation, just walking in general, survival. And insects use a variety of mechanisms to detect sort of those external cues or the space around them in order to make informed orienting decisions. Types of external cues could be sunlight, wind, touch, and chemical compounds. But not all insects are able to detect those external cues in the same way. So flies specifically mostly use eyesight as their sort of mechanism for detecting cues. So they detect both sunlight and polarized light, but they don't just use eyesight. So they also use their halteres, which are modified wings that help while flying, and so they sort of act as stabilizers and do a bunch of other sort of micro things, and they can also detect the Coriolis effect, which is very interesting. They also have specialized hairs called plate hairs, and those exist sort of just below the head. So when the head moves, they're able to detect sort of the pressure and angle of change of those hairs to sort of realign their body to maintain the like head and body position. And finally, some studies have shown that some flies are able to use like mental maps and create mental maps to sort of orient themselves within the space around them. These mechanisms combine to sort of allow for complex movements and navigation. However, not all movements are the same. Here I have a video of sort of a novel form of movement, and make sure you pay attention to, there's a fly here and a fly here, and pay attention to the way in which they remain facing up, but they can still maneuver in all four like cardinal directions. So especially on the left one, you can sort of see it's maintaining its position facing up, but is moving in multiple different directions. So that was a species of fly within the Stenomica genus, which is a super understudied genus, and there are only about 3 known, or like named species within Costa Rica, but there's expected to be a lot more. These two are 2 different species, or we identified them as 2 different species, but we don't actually know which species they are. There's not a lot of research, and they're very complicated to like understand which species, so they could be a completely new, unnamed species, or a part of that 3 that I already mentioned in Costa Rica. And the behavior I showed in the video has only really been documented in a few papers sort of in Europe and dealing with Stenomacra there. But even still, it's just brief, oh, they do weird stuff while walking. But no sort of experiments or any sort of deeper studies on them. So with that sort of background and the observations of the flies moving, I sort of developed 2 central questions to my study. The first is, do Stenomacra species, are they found on certain plant families, and are there certain characteristics within those families that sort of allow for that type of movement? And another one, knowing what we know about flies and the ways in which in the mechanisms that they use to orient themselves, does this novel orientation rely primarily on light? So to answer these, I developed both an observational and experimental component to determine, the observational was used to determine the plant family, position on the leaf, and angle of the leaf. And then the experimental was to test the fly's ability in both the light and the dark. So the observational method consists of me walking around the Simposyum outside, looking on all different types of plants and leaves to identify which plant family they're found on, and then determine if they're on the top, middle, or bottom, and also measure the angle at which the leaf face is sort of oriented. And what I found was that they are only on Heliconiaceae and Marentaceae, primarily on Heliconiaceae. I found about 70 observations or so. They're pretty evenly distributed throughout the leaf, but what's interesting is that they do prefer a leaf that is angled up. So starting at 0, they prefer about like a 60 degrees, almost 70 degree angle of leaf, which is very interesting. For my experimental methods, I had 2 components. I had a trial with direct sunlight and another one in the complete darkness. So I would place the fly in the petri dish over there, and I would rotate the petri dish exactly 90 degrees and chart down the degree the fly is facing, so like up being 0, all the way around. And I would keep doing that for all 360 degrees in 90 degree intervals. And then with the same fly, I would do the exact same thing, but in the complete dark. And at that 90 degree mark, I would shine the light on it to see where it's orienting, turn the light back off, and continue the process. And interestingly, I found no difference between the dark and light. So they're pretty much the exact same in terms of just like facing up in the dark and light. But I did see that there was a trend towards, they're less precise, meaning like the degrees off from like due north is higher in the dark, which does make some sense. So answering sort of these central questions in context of my results. So Stenomacra are found on Heliconiaceae and Marentaceae, positioned relatively evenly, but prefer leaves that are angled up. And the orientation does not rely on light, but may be enhanced with light. So these results raised sort of more questions about Stenomacra and what mechanisms it is using. From my experiment, we know it's not primarily eyesight and using sunlight. They were also not flying, so the halteres don't necessarily help during just like walking. So that sort of leaves the specialized hairs, like the plate hairs, and the mental mapping. But there are sort of some issues with both of these two that don't necessarily fully explain the picture of being the reason for this, or being the mechanism that's allowing this to happen. The plate hairs are more so for about like keeping body position aligned, not necessarily orienting within space. And there's not really enough research to say that all flies use mental mapping, and that it would be complex enough to understand full like 360 rotation or being that precise. So with no clear answer to what mechanism is used for the movement, it is possible that this mechanism is something that is not yet studied. Stenomica could possess a mechanism that is like completely unique to the genus, and would be a very interesting thing to study further. So yes, I'd like to thank these people and all of you people. So, thank you.
There's enough time for questions.
There's not, or there is?
There is.
Okay.
Would you have expected different results if your test spinner was angled at their preferred incidence angle?
I think I would expect it to be less different because they like to move around a lot. So a lot of my trial involved like making sure they were positioned correctly and stopped going on the sides of the petri dish and all that kind of stuff. I think if I had angled it a little bit better, maybe they would have been a little bit more chill, or probably a little bit more precise because it's not such a harsh. Actually, maybe it could be less precise because it's not as harsh of a change in position if it's flat. Yeah.
Do you have any theories about if you would place them on a different kind of plant, would the outcome be different?
I don't know. I don't think so. I had them in a blank petri dish sometimes, and they or just even in the container, they still did the motion of staying completely vertical. Yeah.
Do you think it has something to do with their anatomy that allows them to, their leg anatomy, not specifically their mental mapping, and that allows them to move like that?
I think so. If you look up close when they're moving, the sort of gymnastics that they're doing to maintain complete position, because they don't just remain facing vertical, they remain facing vertical at sort of the rate at which you're rotating it. So I think it definitely has something to do with their movement. Yeah.
I don't know what this is, but how do you make the observations in the dragon's trial?
Yeah, so I have this whole sort of braille system. It's pitch black in that room, so you can't even see your hand. So I rotated the 90 degrees, be able to feel that it's hit the point, and then I'd shine a light really quick to just this way at it at the face. And then the reason I knew it was kind of working correctly is because once the light was on, they would sort of do that last little bit of orientation back towards the complete middle, which is why I'm talking about the precision. So yeah.
Did you ever put the leaf flat and then rotate it?
Yeah, I did put them in flat and they just go kind of crazy. They'd be able to get onto the sides of the petri dish or climb to the top. It made them very uncomfortable to be on a flat surface. Yeah.
Do you or anyone else have any idea what the function of this behavior is?
No, I've seen one of the most ridiculous things I saw was a heliconia leaf, and in the center vein, there was about 15 of them lined up in a traffic jam, just constantly kind of bumping into each other and then backing up, and then just 15 in a row. I saw them mating too, and I'm leaning towards it's some sort of mating behavior or something like that, or I don't know. Honestly, I have no clue. The more I study them, the more it just makes no sense. Yeah.
I'm just curious, did you observe any of those flies flying?
Yeah, they can fly. They would fly in order to position them. If it's faster for them to be vertical by flying, they would fly.
I'm wondering if there is some kind of specialization towards singularities.
In terms of what?
The plants from the singulares, the order of plants,
Okay. Yeah.
Because there are some beetles that are specific when the leaves are low.
Mhm.
So I wonder if those flies maybe are just adapted to this flight and big
Yeah.
leaves, right? And
It's possible.
how they get there.
The species, the ones found in Europe, that was documented, they're also found on sedges, which are not as wide, but they still kind of do that behavior. So it might be that. Right.
I didn't read your paper, but for your final submission, you might check out how other organisms use gravity to orient. This could be an interesting source of ideas in comparison.
Yeah, definitely. I was reading something, and it's interesting because the force of gravity is so small on an insect, especially a fly this size, that their mechanism would have to be like so finely tuned into gravity. But they are able to detect the Coriolis effect using halters, so I'm sure it's possible.
Thank you, Matt.
Thank you.
Focused on this one species, Euglossia viridissima, and their foraging habits when they're looking for fruit bees, and I'll talk about that a little bit. But before we can talk about orchid bees, we got to talk about orchids. So orchids are the most diverse plant family in the world. There are 35,000 species worldwide and 1,500 species in Costa Rica alone. So these are all pictures taken at the Montero de Orchid Garden, and you can just see they're so diverse and amazing, and they're just the coolest. So the cloud forest in particular is a hot spot for orchid diversity, and so that's why this is such a great place to study them. So some of the larger species of orchids are pollinated by a specific group of bees called orchid bees, and it's thought that male Euglossini, which is the tribe of orchid bees, are responsible for the pollination of 10% of neotropical orchids, which is a lot of orchids. Okay, so this is an orchid bee. This is the dudes I was studying. Okay, so what are orchid bees? So orchid bees are in the tribe Euglossini. They're a group of solitary neotropical bees. So that just means they're native to the tropics in the Americas and they don't form hives. So what makes them special? So they're special because of their foraging behaviors. So females forage sort of as you would expect, like most bees do. They collect pollen and nectar for their larvae to eat, and then they build nests out of things like mud and resin. But males, on the other hand, okay, so most of the time with insects, males just have sex and die, and that's all they do. But orchid bees do it in the most extravagant, flamboyant way possible. So the males spend massive amounts of time and energy just flying around, and they collect perfumes, and they store them in their legs right here. And then when they're trying to mate, they'll release all these perfumes like some kind of expensive Old Spice Axe body spray, middle school performance, and hope that a female will come and mate with them, which the female's like really the one, female choice. Okay. Okay, so why this is important? Okay, so in this process of collecting perfumes, there are several orchid species which rely entirely on male euglossum for their pollination. And so you can see, right here, this funky little thing, like attached to this guy's face, is actually an orchid pollinia, which means it's like this little capsule of orchid pollen that's been like super glued to this bee's face while it was collecting perfumes. And then as the bee goes on, it'll like take this little pollen package to like another orchid, and that's how some of these orchid species just do all their pollination. So why are they important? Why should we care? Basically, not only are they responsible for the pollination of these super specialized, amazing orchids, but they're also important for the pollination of several subcanopy and understory plant families. And a lot of other bees pollinate canopy species, but these guys are responsible for a major brunt of subcanopy and understory pollination. So they're really important to the health of like the entire cloud forest. So that's why we should care. Okay, and so my question was, okay, I forgot to tell you guys. Bees can see in ultraviolet light. So like humans, we're basically like, we can't smell or see or hear anything. Like we think we know everything, we don't. The bees know everything. They can see in ultraviolet light. Okay, so I was interested in what visual patterns are present on multivariate orchid species, and that includes ultraviolet patterns. And what role do they play in Euglossa viridissima perfume foraging behavior? Okay, so this really dignified young lady here is trying to look at some orchids with a black light to see if they have any ultraviolet patterns on them. And so I did a lot of that. And so I looked at these 4 locations of both cultivated and wild orchids, and I, using a black light, would look at what sort of patterns are present on the orchids. And I compiled them into this big inventory of 55 species. And then I grouped them into these 4 categories based on what I saw. Okay, so with the help of Eladio and Andres Cruz, we were able to identify all of these species of orchids, right? And using that information, I was also able to search up like what pollinates what. And so basically, I found that there wasn't really a correlation between who pollinates what kinds of orchids in these groups, which was a little discouraging because I'm trying to see like, do orchid bees like these kind of like patterns versus these ones? But I was like, okay, whatever, let's just test it anyway. So then I directly used the 4 categories I found in like live orchid species to make these 4 categories of fake orchids. So I was going to bring them, but I forgot. So sorry guys, but you can see them here. But there's like these 4 treatments of orchid species, or not orchid species, of just like experimental orchids. And they were meant to emulate the patterns that I saw on the live orchids. Okay, so then I set them outside and I baited them with eucalyptus oil, which the orchid bees just love. They're just like, they come running. So I would set them out every morning and I would rotate these like flowers on skewers like every 15 minutes, and I would just like sit there and every few minutes I'd write down how many bees were on each treatment. And it was really hard work. I just like sat in the sun and watched the bugs. It was rough. Yeah, so as you can see here, we've got some bees. We've got 2, I keep forgetting I have this thing. Okay, we have like 2 males right here and here, and then this bigger bee here, you can't see it, but she's got an ovipositor, is a female of the same species, and then there's 1 lone female honeybee here. Okay, so I watched the bees, and this is what I found. So they were kind of into this control, so the all gray, non-reflective, just orchid. liked the all-white orchid, which was like counter-reflective. And then they liked a little bit better the like orchid with the white center, like directing them to the perfume. And then they really, really liked the one with the ultraviolet center, directing them to the perfume. So that's kind of how it went. And so, yeah, something interesting, I'll touch on this in a minute, but most of the orchid bees were males, but there were just like a couple females in there, which was just kind of like a little bit funky, and so we'll get into that. Okay, so the main takeaway here is that Euglassa viridissima bees showed preference for flowers with a central white or fluorescent spot. So they were really into that like bullseye flower, just like showing them where to land on the flower. Okay, so from my orchid inventory, basically there was no relationship between the groupings of the visual cues on the orchids and the pollination strategies, suggesting that there's no co-evolutionary, there's no like co-evolutionary relationship between like these markings and their pollinators, right? And it also, and then it also, because there was no connection between the genus and the groupings of visual cues on the orchids, it's likely there's no like phylogenetic relationship between how an orchid, has these patterns on the orchid and their genus, genera. Okay, cool. So then we get to the cool stuff, the experiment with the bees. So why were there females? I cannot tell you. Like, my theory was that the females were showing up for like the Axe body spray show, right? Because they smelled some nice like eucalyptus oil, and they were like, oh, there's some males. Like I could, lay some eggs. It would be cool. But turns out they like did not care about the males. They actually appeared to be foraging for the perfumes themselves. Like they were on the flowers, like using their mouth parts to like get all up in there, and they just like weren't interested in the males at all. And in fact, both the females and the males would like fight. It was crazy. They would like there would be one on like a flower, and then like some bee would like come in and just like swat at him, or like sometimes they'd like come in from above and like they'd like fight in the air and then they'd like fall down. It's like a soap opera. It was great. But you could definitely do like some research on the interspecific interactions between bees at a foraging site, and like sort of like people have done like coatis and stuff like that, and how they interact when they're trying to eat. Like the bees do the same thing. And again, main takeaway, the bees love the bullseye. So they're like really into that like central white or fluorescent spot on the orchid. Okay, so again, why should you care? Basically, here you can see her ovipositor. Like, look at that stinger. That shit would hurt. Oh, sorry. Anyway, basically, since these guys are so important for like pollination of like a huge variety of plants in the entire understory, and they're so important for like the survival of these like super specific, like ask me about bucket orchids after this, like I will give you anyway. They're like pivotal to the survival of several species of orchids that like otherwise just like would not be pollinated without them. Okay, so basically healthy bees means a healthy cloud forest, and that's why they're important. Okay, so I'd like to thank my advisors, Emmy and Naomi. I'd like to thank Alario and Andres, who basically found all the orchids and identified them. So they did like a lot of work. I'd like to thank my host mom, Marcela Alvaro. Muchas gracias. Te quiero mucho. I'd like to thank all the places that let me look at their orchids, and the Monteverde Institute, and because I know they're probably watching, thanks to my parents. I know my mom sent the link in the family group chat, so I'm sure
some really cool paint. Emmy went really crazy at the craft store. We got some like fluorescent paint that, and I used like none of it, so we have a bunch. So if you want to go play with that, great.
Did you show the photograph, the penultimate slide?
This one?
Yeah. Did you say that B on the left is a female?
Yeah. So this is the female right here.
That actually looks like a tongue.
Well, this is her butt. This is the stinger coming out.
We can look at it another time. Yeah.
Got it.
Thank you, Ella.
Feedlots is a whole separate issue that I'm not going to get into because we don't have time for that. But all cows at least
spend 6 to 10 months of their life in pasture with their mother after they're born, and the cows that are like grass-fed will spend a lot more time at pasture. Basically, all of which is to say is that pasture is really important for the ways that we raise our cattle, and also pasture productivity is really important for the ways that we raise our cattle, because the more grass that you have, the more cow that you get, and then the more money that you get. And generally, farmers like that. That's good. So there's a couple different ways you can go about doing that. One of which is to use fertilizer, which comes with a ton of associated carbon costs, which are boo-boo. We don't like those. Or you can manage your grazing and do rotational grazing and manage basically the way that your manure interacts with the ground and the soil to also get a bunch of nutrients into the soil through that. But for that to happen, the nutrients in the cow dung need to like decompose and get into the soil, because the nutrients are locked up in the organic matter, and so when they're locked up in that form, they're not like available to the roots, and therefore not super helpful. So, like dried out cow patties sitting on top of the soil, not anybody's favorite, not super helpful, not super useful. Cows and methane. These are the environmental issues I was talking about a little earlier. The way that they break down cellulose, which is like grass and stuff that we can't break down because it's really complex, is through enteric fermentation, which also produces a lot of methane, but they mostly burn out, some of which they also fart out, and some of which is in their waste, mainly in the form of like the anaerobic decomposition of the waste, which happens with all organic matter, which is also why you should not put organic matter into landfill, because then it will also do that, and that's that. Yeah. But that is where dung beetles can help. So dung beetles help with a lot of things. They do a ton of ecosystem services. They're very, very cool. So not only do they help, well, okay, back up, beetles, dung beetles, what do? They eat feces, but they also lay their eggs in feces, and they do this in a couple different ways. The family is Scarabeidae, whatever, that one. Sorry. But so there are rollers, which are like the ones that roll those really big balls of dung in that very like National Geographic friendly way that we're like, yes, a dung beetle. So there are those ones, and then there are also tunnelers, which basically like dig a tunnel through the poop into the ground beneath it, so they have like all of these little balls of poop in their tunnels, which they lay their eggs in. And then there's the lazy ones that just like hang out in the dung the whole time, and I'm sure they do something helpful. Yeah. So through this, they like help aid to get the nutrients underground through their tunneling and also like their burying of the dung balls and all of that stuff, and aid in decomposition in that way. They also aerate the manure, leading to aerobic decomposition, which doesn't we like. And also they just aid in generalized waste cleanup. If we didn't have dung beetles, like everywhere would be covered in poop, and that would be gross, and we wouldn't like that very much. Okay, ivermectin, that should be capital. Ivermectin is an antiparasitic medication that is not used to treat COVID. Surprise. It's an antiparasitic medication. And basically like gets rid of worms and other insects. You can use it topically, that's called like pour-on treatment, for like lice and stuff like that. Well, you can and also you can inject it for internal parasites. And the way that it works is like it basically messes with the like nerve and muscle communication in cells and kills the animals in that way. So it's handy in terms of like veterinary practices, but it can also have like rolling impacts into other stuff, which is what I decided to do my project on. The thing that I was interested in was to see if the use of ivermectin in cows had an impact on the decomposition of cow manure, like if the residual trace amounts of ivermectin in cow manure would affect decomposition, especially in terms of like aerial lost or disturbed, and instances of dung beetle disturbances. So the way that I did this was I basically just collected samples of manure and like observed the decomposition process, and took photos. And so I collected the samples from Jesus Suarez's farm. So I took samples of cows treated with ivermectin, not treated with ivermectin there, and some from the Sarasakis farm, which were not treated with ivermectin. These are the cows treated with ivermectin. I gathered those samples from concrete that might have an impact, I don't know. And these ones I gathered from the pasture, and those are Jesus Suarez's farms, not treated with ivermectin. The previous slide is Sarasakis farms. This is how I laid out my samples. Basically, I just like plop them down made them into patties. That was gross. That were around like 11 to 13 centimeters in diameter. Or sorry, whatever the one is that goes all the way through. And set them up this way. So the S samples are the ones from Serostechni's farm with no ivermectin. The N samples are the ones from Pizza Suarez's farm with no ivermectin. And the I samples are the ones treated with ivermectin from Pizza Suarez's farm. I also put milk crates over them because I didn't want to deal with turkey vultures eating the dung, which apparently they do. And also when I was laying out the samples, one was like harassing me and like swooping over my head and it was very scary. Dung beetles disturbances. So these were the, sorry, poop. These were, I referred to these as sort of like dig outs and these as holes, and I counted any like instances of these that I saw as instances of dung beetle disturbance. So this counts as 3, this counts as 2. That's how I tallied those. And then the way that I calculated area lost was I used image J and the way that everybody else has been talking about, measuring irregular areas, and then it like gave me a big old list of all the things, and I added them up. I did math. So these were the samples on day 0 when I like first set them up. As you can see, they're all sort of similar size and all similar color, and these are them on day 9. It's kind of hard to see. These are kind of the only ones that like, you're like, oh yes, that is a cow patty on the ground. I can see that. These ones, less so. These had like a sort of medium amount of disturbance on all samples, and then this one was, these were really interesting. One of these samples like decomposed the most out of all of them. Like I don't think if you didn't know what you were looking for that you would recognize it, but the other 2 samples remained fairly intact. These ones are just like, sample, okay, we're going fast. So this is the area lost in square centimeters. On this side, and these are the 3 different treatments, so I have the green dot to represent each one of the individual amounts of area loss per sample, and then the brown triangle is the average. Super high standard deviation on the S samples, like I talked about earlier. I didn't know the test on them, so they were not statistically significant. We're going to get more into that later. And then these were the instances of dung beetle disturbances. So on the Stuckey samples, the S samples, I counted 10 instances of dung beetle disturbance. On the N samples, I counted 4 instances of dung beetle disturbance, and on the I samples, I counted only 1. But also they were, I ran another test, and they still were not statistically significant. Okay, there were definitely some uncontrolled variables, like obviously between the Stuckey samples and the Suarez samples in terms of feed treatment and just like the breed and purpose of the cows on each farm. And then also on the Suarez farm, the surface of collection varied. That like could maybe have impacts of like pollinization in the early hours before I collected them, as well as the feed from the previous day. The reason why the I samples are so much darker and more of a brown color and like turn to brown color a lot faster is because I think the cows were being fed hay in the day before I collected the samples instead of grass. We were eating grass, so it was a lot more green. Yeah, and okay, while none of the data was statistically significant on the things, I still believe that there is a relationship between the two, just because of the relationship that I saw, because there was a difference in instances of dung beetle disturbance and area lost. I had a really small sample size, and I think that I would just super recommend to redo this experiment. Luckily, there's cow poop everywhere and a lot in Davis, so I'm going to do it again with a bigger sample size, hopefully, and more controlled variables, and just to see if there actually is the relationship that I still think that there is because of kind of the data, but also kind of not the data. Yeah. And thank you to everybody who helped me with this project. Thank you to Fern and the kids out there. They drove me everywhere. It was awesome. Yeah.
Do you think that the ivermectin also might be having an impact on other microfauna in the soil?
Yes. I was actually talking to Naomi about that earlier today, because I don't think that the image J area lost would have dung beetle traps. But I was like, I'm interested in dung beetles, but I'm also really interested to see what the impacts of ivermectin are on all of the other types of insects, because I also, like, when I was staring at them, there were a lot of, they looked like beetles. I don't know what they were, because they were like this big, crawling around on the samples, and I think that, like, decomposition is such a complex process that involves a lot of different kinds of life, and I want to see how ivermectin affects all of those. So I do think that there is an impact. Dung beetles were just, like, very clear thing. I was like, that's a dung beetle.
If you did this study again in Davis, do you think you'd expect to see different results?
I think the rate of decomposition would be a lot slower just given the climate. Davis was a lot drier, I mean depending on season, but a lot drier and cooler than here. Decomposition is sped up by like warmer and slightly moisterish environments. I think that what I'm hoping to see in Davis with this is like basically this similar results, but just like on a bigger scale, and hopefully that will lead to statistically significant data.
How close were the patties that you made to each other, like the differences in them?
They were like a meter apart. I would have liked to have them be a little bit farther, but I wanted to make sure that they were like having the same environmental things, and I wanted to have them set up at the station so I could like be close to them and keep an eye on them. There just wasn't that many like flat surfaces that were appropriate to have a lot of cow poop in. Yeah, they were kind of close to each other.
Thank you, Eliza.
Because it affects the liquid substance you've been drinking all day to be able to listen to all these beautiful presentations. So it affects coffee brewers, and how it affects coffee farms. Thank you, Dan.
So my presentation is on the coffee berry borer and the preference of mature coffee fruit. To start off, drink coffee is one of the most important cultural practices spanning back to 15th century Ethiopia, in where coffee was prepared similarly to how we drink it now. Coffee is now one of the second most consumed beverages in the world, and with over 2.25 billion cups of coffee and 1 billion people drinking coffee each day. To supply this, we have 44 countries that produce coffee with 12.5 million coffee farms, 125 million people employed, bringing in $83 billion in the coffee industry. Costa Rican coffee is projected to bring in 1.4 million 60-kilogram bags. It's like those big bags that we saw at Café Monteverde. 1.4 million bags next year. And that is prominent because Costa Rica has a significant place in the coffee industry with 50,000 individually owned coffee farms. 90% of them are on less than 5 acres of farmland. It produces 1% of the world's coffee, and for Costa Rica, it's the third most important export after bananas and pineapple. Since last year, there's a 15% increase in production of coffee, bringing in $372 million to Costa Rica. But Costa Rica, in fact, is also the #1 user in pesticides in the world, using 22.34 kilograms of pesticides per hectare. So I decided to focus my study at Café Monteverde specifically because, unlike conventional farms, Finca Life doesn't use harmful insecticides like endosulfan, which has 58 active ingredients that can cause cancer, which most of them, or all of them actually, are banned by the EU. Secondly, they also treat plants with chemical herbicides and fertilizers, which makes these plants more vulnerable to diseases and pathogens, making them weaker overall. Finca Life, on the other hand, uses accessible and natural agroecological biocontrol methods of reducing weeds and pests. This includes, actually let me explain. So biocontrol, if you're not familiar, biological control, is the control of pests by the introduction of various natural methods. This may be natural enemies, predators, parasites, pathogens. It could also be natural substances like plants, algae, and fungi, or chemical mediators like pheromones. This is all in an effort to disrupt the pest infestation cycle. I think Life, in particular, they use biocontrol methods like intercropping, shade trees, windbreaks, and protecting and promoting wildlife. They do this because there's wildlife living naturally in these forests. So when you intercrop, having citrus and banana plants, allowing for a natural forest environment and allowing for things like wasps and ants and birds and spiders to eat pests that are naturally there. Another thing that they use is a fungicide, a natural fungicide actually. It's found in soils worldwide, and it's Bavaria Vicina, and they use this to spray on all of their berries. So this is what the study sites look like. We've all been there, but you see how we have intercropping, shade trees, banana trees in the back, and surrounded by forests, really making it seem like a forest and not a farm. Yeah, so let's get into what we're actually talking about, the coffee berry borer. So the coffee berry borer is Hypothenemus hempi, or Broca de café. It's basically what you see here is the berries. If you walk around most conventional or most farms, you would see these berries with little tiny holes in the floral disc of them. These are where the colonizing females will dig into the berry and dig their galleries, eating all of the seeds inside and the endosperm. And they're actually one of the only beetles that can digest the caffeine, because caffeine is usually a deterrent for insects, not these beetles though. So you'd walk around Finca Life and you'd find these beetles. In one of these beetles, you can find their entire lifespan. So I would cut open one and you would find from larva to adult female beetle. It is also important to recognize that female beetles are the only ones that dig into the beans. Like Ella said, the males are basically insignificant and they're pretty tiny, so it's very easily distinguishable which ones are which. And in order to get rid of these pests, we need to learn about their behaviors and biology. Especially in biocontrol, you need to know what you're working with to make methods to get rid of them. So this leads me to my question. Does the coffee berry borer have a preference of ripeness in the Café Arabica berry? And how can we better manage the timing of biocontrol methods using this information? And my prediction, talking with the people at Finca Life Monteverde, like Eddie, we predicted that weevils will be most abundant in least mature green berries compared to more mature red berries. This was more or less my prediction and his prediction. I took his word for it because that's what we saw. I didn't really know, and it was a good thing because once I started collecting berries, we were able to see this. So for my methods, I went through 4 steps. Collect, find berries which weevils have dug into. Dissect, isolate weevils from the seed. Infect, set up preference trials using 4 levels of maturation of the fruit. And inspect and check which of the berries they have dug into. So this is what our trials kind of look like, my trials kind of look like. You would find a berry that was dug into, I would dissect them, isolate them by themselves, and then set up choice trials with least mature green berry, a little bit more mature, a little bit more mature, and then red berries. Shut up. Shut up. So okay, yeah. So after 101 trials that I did, spanning over 5 days, I was able to find with significant confidence that these berries, or these weevils, preferred red mature berries. In this graph, I had maturation on the bottom showing green unripe, half green, half yellow, half red, and red mature, and comparing to the number of weevils present in the berries. So this was surprising. We did not expect this, and I was happily surprised because this meant that we can move on with our conclusions, basically. So in order to create effective and efficient methods of biocontrol, we need a variety of biocontrol methods to show success. Studies have shown that these weevils are attracted to pheromones like ethanol and methanol, which makes sense why they would prefer more mature red berries, because these are ripening pheromones. One of the biocontrol methods used is that they use lure traps. During their flying season of March through May, they would fly into these and die, basically, which is a great thing because we don't want them in our berries. Another form is fungicides. With this conclusion, I can tell Finca Life that they should be spraying their fungicides more often, because not only do they infect just green berries, but they also infect a variety of them. We see that they infect from every single form. When you go on the plant, you would see a variety of maturation from green to red, showing that they will have no preference, but very much a preference to what they like. So, in conclusion, as climate change, so most of the research done on coffeeberry borers was done more than 20 years ago. So there needs to be an update on the biology and behavior of these weevils in order to find effective forms of biocontrol. Further research may look into sterilization methods of weevils or finding natural repellents that these farmers can use. In conclusion, as climate change is creating more variable and extreme weather and temperature, it's allowing for more pests to rise. It is important to research the ways that our agricultural industry will change and make sure that we're protecting these farmers that aren't using conventional pesticides. And we need to protect and create natural methods to reduce these pests and protect the coffee in the world. Thank you. Gracias a Emilia, Naomi, Café Monteverde, Guillermo, and Eddie, the biocontrol manager, and Wilbur for driving me to Finca when Emmy, thank you. Y hola, Naomi.
Oh. There is enough time for questions.
Yes.
Yeah, you said coffee is the second most consumed beverage.
After water. Yes.
I have a question. Did you talk to them about, or anyone at the farm, at any point about how it's changed? You said it hasn't been studied for 20 years. But, like, I remember as a child, they used to say you had to pick all the red berries because that was the only one that was being affected. According to your studies, now it's going into green berries.
Right.
So they're probably, like, adapting like
Yeah.
any other animal would
Absolutely.
do what we're doing. So did you talk to them about that?
Yeah. So talking with the biocontrol managers there, they informed me that they thought, and most of the people thought, I talked to multiple people there, that they infect majority green berries. And that's what I took their word for it, is because it seemed right. But this is also because, again, because of climate change, we're seeing multiple harvesting seasons per year. We're seeing, instead of one harvesting season, we're seeing 2, 3, and 4. So we're having a variety of times you have to pass through the plant to collect every single berry of different maturations. Yeah.
When you find one red color berry, can you know that
was infested when it was red or when it used to be green?
That's a good question. So my hypothesis was that a green berry would be more viable because it gives them more time for them to mature inside the berry. A red berry, I would agree, it wouldn't make sense for them to dig into a red berry because in a short amount of time it would be like, it would just turn gross and they wouldn't be able to thrive in it. They take about 8 to 12 hours to dig into the berries, and it really depends. Like each berry I would open up, you could see varying forms of how much they dug into the seed. Sometimes they would be completely gone and there'd be 5 weevils inside, or sometimes it would be just like they just nibbled on the seed and then that was it. It was varying. Yeah.
If they're in the berry, is that berry basically unusable?
Yeah, so they, in bad years, they can affect 40 to 50% of the yield. And that really messes up small farms like these because most of their profit just goes away. Yeah, Jake.
Do you know if there's a different amount of caffeine in the matureness of the berry, and at what point would it be like too much caffeine for them to tolerate?
That's a good question. I read studies that showed that they have zero effect to caffeine. They actually have gut microbiomes that can digest the caffeine inside the berries, which is one of the only beetles that can do this. That's why they're so prevalent in these seeds themselves. But they have looked into using caffeine as a deterrent even to kill them off. But they've just gotten used to it and developed more gut microbiomes and stayed alive. Yeah.
Thank you, Dr.
station. The bad news is I'm going to give a speech for about an hour. So we can have a quick break right there. I'm going to take myself
¿Va a salir o lo paro? ¿Ah? Okay.
It's about that time. We have about maybe 85 hours left in Monteverde, in case you're wondering. So